Hello, everyone can hear my voice? We are waiting the speaker. Um, okay, he asked me where is the link. Yes. All right. Okay. Once the speaker get in, and then we can, I think, just start the session. I think he's now logged into the system. All right. Hello, Dr. Azwan. Can you get my voice clearly? I think your microphone is off.
Let me see. How is it now?
Okay. It's clear. All right.
Alright.
Okay. Okay. I think welcome, Dr. Azwan. We in the program. I
Hello, Pak Cahyo, you are free.
Dr. Chayo, you are freezing.
all right. Okay. I think looks okay. All right. Okay. Let's start the session. Okay. Thank you, everyone. Assalamualaikum warahmatullahi wabarakatuh. Welcome back, everyone, to the summer school program of Department of Animal Product and Technology. For this session, today's sessions, again, I will chair. It's my honor to chair the sessions. I'm Cahyo Budiman. Let me share the program today. Just here. All right. Can everyone see my slide?
Yep.
Okay. So for today, the fifth day, if I'm not mistaken, yeah, we have lecture 9 and lecture 10, and in the middle we have virtual excursion to animal product processing industry, and also overview of Indonesia culture from Yogyakarta. The lecture 9 supposedly Professor Dr. Irma Isnawi Arif from IPB University, followed by lecture 10 from Professor Dr. Azwan Awang from Faculty of Sustainable Agriculture, University Malaysia Sabah. However, due to circumstances, we swapped the schedule. So Prof. Azwan will deliver the talk first, and then Prof. Irma will be in the lecture 10. But I think Prof. Irma actually is ready now also. So, but anyway, since we swapped already, so the first lecture for today, the lecture 10 will be by Dr. Azwan, from Faculty of Agriculture, University of Malaysia, Sabah. We'll talk about proteomics for agriculture. I think Dr. Arjun is already with us here. So before we start it, let me introduce briefly about our speaker, Associate Professor Dr. Azwan Awang. I think the committee made mistake on the names. It's supposedly Azwan Awang instead of Azwang Awang. We'll talk on proteomics in agriculture. Okay, can see this. Dr. Azwan is from Faculty of Sustainable Agriculture, University Malaysia Sabah. And sorry, if you see the map here, Sabah is located somewhere not Borneo, not Kalimantan. Yeah, it's quite close to Indonesia. And then when the capital city of Jakarta really moved to the North Kalimantan, so Sandakan will be very, very close to the capital city of Indonesia, but we don't know whether it will come true or not. Anyway, he's educated in all in Japan, BSc, and then in Niigata University, follow with his master study in 2002 in Niigata University also, and 2010 in Niigata University. And his expertise is in proteomic analysis for plant proteins. Dr. Azwan's research experience started as a senior research officer in Center for Cocoa Biotechnology Research, Malaysia, Cocoa Board, Malaysia. And then after that, he became researcher in Japan Environmental. Before that, he was researcher in Japan Environmental Technology, Niigata, Japan. And then after completing his term as senior research officer, he joined University Malaysia Sabah as associate professor. He is very expert in proteomics techniques, particularly on 2D gel and then mass spectrometry, and also non-gel, free gel based of spectrometry. Then there is member of Genetic Society of Malaysia and also International Plant Botanical Organization. He do a lot of research. He received several grants for regarding the research on his field in proteomic, as you can see here, mostly on the plant proteomics. I think without further ado, I would like to invite Dr. Azwan to the stage and start the presentations. But before start, I would like to deliver some housekeeping information for the students. I think supposedly we have more than 150 participants as usual, but I think it's now keep coming. And then students, please ensure always turn on your video to ensure that your physical presence during the program, during the class, and then also ensure that you fill in the presence forms or registration form that will be delivered by committee during the talk. Yeah? It will be 2 times, earlier in the early of the talk and then at the end of the talk. Yeah? So you should have the committee informed you that you have to register twice in order to get the complete credit of this course. And then for those who has questions, I would like to also allow you welcome your questions either in written forms, or you may ask the questions directly to the speakers. Yeah? Just raise your hand, and then I will let you. The committee will turn on your microphone, and then allow you to discuss directly with the speakers. Okay, Dr. Azwan, we have actually 1 hour 40 minutes, but I hope that you can allocate about 20 minutes or 30 minutes for the discussion with the students after you talk.
Sure.
All right. Are you ready? I think you will present from your own computer, yeah?
Yep.
Okay. Please proceed.
What should I do? I should share screen, is it?
You can click the share screen.
All right.
Okay.
Okay. Sorry. This is my first time using Zoom for presentation. Usually we use Google Meet. All right. Let me see.
Sorry, I think your voice is not clear enough. Maybe you have to increase the either volume or your.
Sorry, the resolution is not good. I have problem with the resolution. Is it?
Can everyone get the Dr. Azwan voice clearly?
Not quite.
Not
enough.
It's clear, but it's not really loud. It's not clear
I
enough.
see. How about this?
That's better. That's better.
Okay, all right. Probably.
Speak a bit louder.
Here's the thing, my Mac battery died, and I have to use my Windows now. So all right,
All right.
let's
Okay.
see.
Please proceed, Pak.
Okay, all right. Assalamualaikum warahmatullahi wabarakatuh, and very good afternoon. How is everybody? Good,
Good.
good. All right. How about the participants? Can you write somewhere there? You ready? How's everything? Good. Can I see the chat there? Can write somewhere there? Yes. Awesome. All right.
Someone
Okay.
said not very good.
All right. Okay. Let's see. All right. Okay. Thank you very much for the opportunities from IPB University. I would like to thank you to all the professors and doctors from IPB, and especially to Ibu Irma. Thank you very much. All right. I really value the opportunity given, and I am really excited to meet all the future young scientists. Right?
Thank you, Professor.
Yeah. Um, if anybody would like to ask questions, especially the students, you just can write somewhere in the chat room. We try to entertain you guys, huh? All right. So my name is Azwan, all right, and the photos there is my faculties. That's the only probably the beautiful place you can see. The others are farms, right? So we would like to talk about today about proteomics, right, and how proteomics can be used in agriculture, right? Right, as being introduced by Dr. Chayo, all right, I'm in the Borneo here, and in the most east of Borneo Sabah here, right? This is the only faculty from University Malaysia Sabah, and we are alone here as usual as agriculture faculties, right? And I've been informed that not only most of the participants are from Indonesia, and I also know that they are from other countries as well, right? Which countries are the farthest from our region? Anybody can write in the chat room there.
Dr. Azwan, sorry to interrupt.
Yep.
We got some feedback voice from your side. Maybe, do you use two computers?
Sorry?
Are you using two computers now? I mean,
no,
there is a
I'm
feedback
just
voice.
using one computer.
All right, okay. All right,
is there
then.
any... Hold on, let me see. I'm only using one computer.
Okay, Pak, please proceed, Pak.
All right, okay, all right. Okay, so it is my hope that by the end of these sharing sessions that attendees can explain why proteomics is important and how we can describe proteomics, right? Secondly, by the end of our lectures, you can describe the fundamentals and the principles of the techniques that have been used in proteomics and some of the core technologies. And finally, it is our hope that we can highlight or provide examples, some of the applications of proteomics in agricultural research. Yeah. So we will check later on whether we can achieve all these objectives, right? All right. So for the content, right, we will talk. Well, we'll categorize into three parts, right? We talk about the definitions of proteomics and then why we so much care about proteomics. Secondly, we will talk about what are the techniques and the technology, right? And finally, on the applications, right? Anybody of you have heard at least what is proteomics? Where is our chat room? Can't see our chat now. Uh-huh. Okay, so let's go for introduction. Yeah. All right. So as we all know that proteins are large macromolecules that are made by amino acids. Right. It's a long chain with plenty of amino acids. All right. And we have 20 amino acids. So we can select whichever amino acids that can be combined in proteins. And proteins have structures. Because they have structures, they can do many functions. Not like DNA. They only have that double helix, so they don't have that structure that can make them functional. All right, like airplane, right? We have structures, we have wings. That's why plane can fly. Same as protein as well. Protein has structures, so that's why they have different functions. So proteins are the some people call as the molecular robots. They're doing all most of the work in the cells, right? They are also the postman, sending messages, right? They are mostly enzymes, okay, catalyzing reactions, right? Some of them work to prevent pathogens, right? And they also, as a courier, transporting molecules from one place to another place. They also as a housekeeper, right? Some proteins like storage proteins are the it's like a bank, right? They become a nutrient for seeds to grow, and we all know that proteins also is very strong, right? Something like keratins, that like our hair is made by keratins, and the same keratins is made also like the horn of rhinoceros. Rhinoceros horn is very strong. That's the same keratin made by like our hair. All right. So all these are proteins and they're very important, right? Okay. So we go to the definitions of the proteomics. Start with the word proteome, right? So we have our genome, right? Genes, which is in the our nucleus, and also if plants in the chloroplast and also mitochondria, right? So the whole proteins that being expressed in a tissue of cells, that what we call as proteome, right? Coming from the word proteins, and ome is like a whole world, right? And proteomic is just a study of the proteome, right? Professor Mark Wilkin from Australia just coined it in 1996. 1996 is where I just joined the university as a freshman, right? So that is proteomics. All right. So do we have any questions? No? All right. So we can continue. Yeah. So proteomics is a study of proteome. So whatever proteins inside a cell tissue, that is what we call as proteome. All right. So let's see here. Everybody knows that all the genetic potentials are stored in DNA, right? And protein is just like instruments, like we just talked right now. We have something, yeah? Right? Okay. So um, from DNA, right, that is the where the information stored. It must be expressed into protein. So protein do all the work in cells, right? And it has to go through process of transcriptions and later on translations, right? And protein itself sometimes been modified, so it becomes can have different functions depend on situation and needs, right? So DNA is just genetic potential here, and the people that have been doing all the most of the works are the proteins, right? So if you look here, all right, so DNA is the plan. We have proteins, where are all the real workers, and mRNA is just like the engineers who read DNAs and give it instruction to proteins, right? So proteins are the real workers, right? And that's why they're very important. So when we want to understand a biological system, let's say we want to understand why plants grow, right? We want to understand why certain animals behave differently when they have some diseases, right? So we are trying to understand a biological system.
Dr. Azwan,
So we want to understand, yes, sorry?
sorry to interrupt again. Some participants inform me that they have a problem to get your voice clearly. The feedback and not clear yet. Maybe it will be better if you use the headphone microphone if you have, and then is your voice is broken from for some participant also. I think some. Do you have any microphone or something?
Microphone. My microphone is embedded with the camera.
All right. I think so. For the participant who cannot get clearly, I think.
Hold on. So you have problem with with the voice. Hold on. Let's see if I have another options here.
There are some glitch, Pak.
Hold on. Is it possible if I try to switch to my iPad if the sounds?
All right. Okay.
Would that be possible?
I think that will be better. Whatever can improve the.
Okay. Let me drop first. Yeah.
Okay. All right. Okay. So everyone, Dr. Azwan will off first, and then we'll be re-login using another device.
Hold on. Yeah.
Okay. Okay. While waiting, so if for those who has already the questions for his talk, you can start. You can put it on the chat room or you can either in the public chat room, or you can chat me privately, whichever convenience for you.
Hello.
Hello.
Achayo, can you listen to me? Can you hear me?
Yes. It sounds better, but
Sounds better.
how about the others? Students? Anyone? Better, sir?
Better.
Okay.
That's good. Yeah, the guy
All right.
with the
Okay,
goat.
good.
Awesome. All right. Screen. All right.
Just maybe a bit louder, but
A bit louder.
yeah, that's just nice.
All right.
It takes
Hold on.
some time to travel from Sandakan to another part of the.
All right.
All right. Okay.
I'm trying to share content. Screen. Hold on. And now I do not know how to share. Can you see now? Anything you say? Can you see?
Are you using iPad?
I'm using my iPad.
Yeah, you can share, and then you have to go to the home screen, I guess.
I have to go to home screen, share content, right?
Yes.
And then
will display all your home screen.
No, I can't see anything.
File that you want to open.
Screen photos. All right. Screen recording. No. How about now? Can you see?
Yes.
You can
Then you
see
go
now,
to the file that you want to share. Yes, correct. Okay.
All right, good. Awesome. All right. So we talk about the definition of proteomics and why proteins are very important, because they are doing their work. Means that all the works at molecular levels, most of them have been done by proteins. People talking about DNA, that's only a blueprint, and then people talk about messenger RNA, those are only intermediates. In agriculture, these are what we call as middlemen, right? They can change everything there and will result in something else at the end, right? So that's how important is proteins, and protein as well. So for them to have different function, to make variation in their functions, they can have what we call as post-functional modifications. So they can do extra, they can have extra functions. Something like us, we are dressing like this. If you want to go to the sea, or you want to go dive, then we change our gears, and then we can do extra activities. All right. All right. So when we want to understand biological system, we want to understand probably how the nature works, right? How the plants are growing, why certain plants are responding differently on the applications of fertilizers, why certain livestock, probably cattle, behave differently on sand management. So we want to understand this biological system. We can of course um, we can analyze or see phenotypes, but we can look as well at the omics. We can see at the DNA levels, right? What we call it genomics. This, what you see is what can happen, right? Then you can see as transcriptomic and proteomic, metabolomics. All these three are the product of genomics. Genomics is just the blueprints, and the others are the products of genomics. While transcriptomics, what we can see is just like what appears to be happening, but in proteomics, what makes it happens, right? And as well, the down below there, which is very near the phenotypes, is metabolomic, is what we can actually, what has happened and what is happening. If we just study on genomics, if you remember your genetic about penetrance and expressivities, that sometimes that a certain organism may have the genes, but it's just that they don't express, or probably they express the genes, can be shown as phenotypes, but they have different levels, right? Something like the extra digits of our thumbs, right? We all have that genes, right? But not all of us have extra digits for our thumbs. And even though we have, some people have extra digits for our thumb, for the thumb, they have different levels. Some people can move it, some people can don't. If you see the genomics, you just see that, oh, people have extra terms, extra digits, but not necessarily when you look at phenotypes, right? That's why there are many levels that we can analyze if you want to understand the how natures work, and of course, not any singles of these approaches can cover everything. I do believe that because we are all complicated, right? That's why we have to look at all of the stages, right? Combining everything to understand. Probably still yet we probably difficult to understand, but we are trying to understand by different levels. All right. Right. So what are the advantages of proteomics over the others, right? So we know that proteins are the molecular tools for most cellular functions. So if we get the workers, so we can understand probably what are the mechanism of certain functions, right? And then proteins are directly related to function, yes, right? There can be modification in the proteins, right? Some proteins can have some modifications. And importantly, if we want to talk about transcriptomics, sometimes mRNA that been transcribed are not the correlations between transcribed and translated proteins sometimes are not that well correlated, right? Maybe you can see that the mRNA being transcribed a lot, but if you look at the proteins, sometimes the proteins are maybe they have little amount or different type of variants that if you compare with the transcript, right? And then, of course, some scientists says that metabolites are the closest to the phenotypes. Well, we agree with that, but well, currently, people are doing metabolomics still have some challenges whereby when they want to identify what are the metabolites, they cannot determine because of the lack of a viable database in the publics, right? So these are some few things that proteomics are superior compared to other approaches. All right. So different type of proteomics and different type of applications, right? Don't worry about all those terms. It's just that if we want to understand what are the proteins that actually working in certain conditions, on certain times, on certain environments, that we have to first profile the proteins according to what we desire, we want to know, right? And probably if you want to compare after you profile all the proteins, probably you want to compare with other conditions of the same cells or different cells, you can do that, right? So there are plenty aspect that proteomics can be applied, but in this lecture, what we will cover mostly on the profiling, and then we want to see how proteomics can be used to differentiate between different conditions of cells and tissues. All right, so I stop here first, right, before we go into our second part, all right? Is there any comment or questions from the students?
Students, if you have any questions so far? Not yet. All right. Okay.
Not yet. Can I give questions?
There is one question.
All right. Okay.
Can you read from the chat, or I think I'll go read by myself? Oh, Raka, are you there?
Oh, this is very
okay. The
difficult.
mic is still blocked. Pak Arman mungkin bisa di on-kan mikrofonnya ya. Okay, I will read the questions first. I'd like to ask some information about prion and how the strand protein is able to cause a lot of damage to human and animal.
Wow, this is very, very difficult to me. This is not my subject matter. I am sorry. I am a plant scientist. I don't know why. do you know, Dr. Chayo? Can you help me with that?
It's your session, Pak, not mine. I don't know.
All right.
Okay, we'll keep it at the end. Maybe we can discuss more about this.
All right. Okay. Of course, things that should be explained is that I am a plant scientist, and then some of the topics probably later on, especially on the applications, I will just share, right, and how proteomics can be used. Probably subject matters, probably people can help me later on, right? Okay, let's see. All right, if there's no questions, I have to again. All right. Start broadcasting. All right. All right. Okay, so now we do understand that proteins are the workers in the at the molecular levels, right? So if you want to understand how certain certain mechanism, or let's say how plants react on certain stimulus, so we want to understand at the protein levels, right? What are the techniques and what are technologies that we can use or are used in proteomics, right? All right. So these are the general workflow in proteomics. Which whatever proteomics it is, these four is the basics. Number one, you need to isolate the proteins. Number two, when you isolate the proteins, remember in the cells there are plenty of like thousands of proteins. Now you isolate, right? Now you have in your hands different type of proteins. So you want to see which one that you will analyze. You need to separate, right? You can separate based on the characteristics, which will later on we will discuss about that, all right? And then after you separate, you need to detect because you can't see protein with your eyes, right? And then later on, if you find something that interested you, you need to identify, right? So these are the four steps in proteomics. All right. So many people like to use different terms, right? Some technologies in proteomics. What we will cover in this lecture is the gel-based proteomics, what they call it, which is using either SDS page or 2D page. And another one is what we call as gel-free proteomics, right? In gel-free proteomics, I will also include on the use of isobaric pack, where we can quantify relatively, right, proteins by using mass spectrometer. I'm sorry. All right. Okay, and then I would like to show here, all right? They have several generations. It's based on, well, as years goes by, the technologies or the strategies or the techniques are getting better, right? These two DE or electrophoresis or gel-based proteomic, what they call as first generations, and people start using later on taggings, they call second generations. They have this gel-free proteomics, they call third generation. And the fourth generation, they say that if you really knows what proteins that you want to see, you can actually only monitor that specific proteins that you want to see in different type of sample, what they call it as fourth generations. All right. Okay. By the way, if anybody have questions, I cannot see because I'm using my iPad. I cannot see the notifications.
Oh, no worry, no worry. I will collect all the questions, and at the end, actually, usually we put the questions at the end of the talk, and students will ask directly.
All right.
You will have many questions, but don't worry.
All right. Okay. I just feel lonely talking alone.
Okay.
All right. Okay. Now let's go for the first one, what we call as gel-based proteomics. This is classic. This is the basic of all proteomics, right? They call it gel because you're using a gel called acrylamide, right? We look at later on, right? So in these techniques, they have what we call as one dimension or two dimensions, right? And this, many people that really like technologies in science, they call it, well, classic, so it's not useful. No, it's still valid and powerful. I will show you later on that the applications that I will share are very current papers, right? And however, classic proteomics using gel have certain disadvantages, of course, right? But it's very superior if you have your samples of proteomes, which is low in complexity, or you already fraction it, and you combine with shotgun or gel-free proteomics. That will be very, very powerful. All right, let's see that later on. All right, that is a cell. Let's say it's that. We're going to the technique now, and please follow me. All right. So we have that cells, right? Looks like it is happy, right? In the cells, of course, you have organelles, compartments, and we have proteins like all over the places. They are in the cytoplasms, they're in different organelles, they are integrated in the membrane. So what you see there, the one with the colors with the like triangle or square, those are different type of proteins that you can see in these cells. Right. Of course, in the cell as well, you have other non-proteinaceous molecules, right? So remember, the first step in proteomics is for you to isolate the proteins. Now, after you isolate, right? How do you isolate? Usually you will, for example, for samples like plants, you have to grind because the plant cells have cell wall, but not for, for animal cells, which is relatively easier, right? Of course, you need to grind, but not that much power. It means that sometimes you can use sonification as well, right? So you isolate your proteins and whatever molecules that will come together, right? And after that, you have to purify your proteins, right? You purify your proteins, either you precipitate them or you're probably based on polarity, probably they will be in different phases, right? So it's up to you, right? So whichever that suits your needs, right? Now next, remember that we need to do separations, right? So now how you have, let's say like hundreds of proteins now and you want to profile them. Imagine as well that you are in a classroom, right? Let's say you have 60 or 70 people in the classrooms, right? They are together and you want to know how many boys and how many girls in that 60 or 70 groups. So if they are cluttered together, it's very difficult for you to come, right? But if you can separate them, that is very pretty easier for you to do analyze. Same as well with proteins. So now you have like hundreds of proteins for you to analyze what type of proteins are inside your sample. So the best way is for you to separate. So the very basic characteristics for separation is based on size, right? Different proteins have different size, right? So we have very large proteins until smallest proteins. So with this, we use a gel. We call it sodium dodecyl sulfate polyacrylamide gel electrophoresis, SDSPH. Okay, many of you, I know that you know these techniques. Right? Electrophoresis is a technique that whereby molecules will move, molecule with charge will move towards, let's say, from positive to negative or negative to positive in an aqueous media, right? When you put some charge there, right? Electric current, sorry. So you have your gel made by acrylamide and bisacrylamide. So acrylamide and bisacrylamide is like making a pore, right? It's like your coffee filters, right? You can see that they have pores there, right? So that depends on the percentage of acrylamide that you put in. If you put a little acrylamide, means that your pores will be big, means that many big size protein will go through. But if you increase your acrylamide, that your pore will become smaller, means that big proteins will having hard shape to go through, while small protein will pass through easily, right? So your protein first will be added charge, all right? That's come from the detergent called SDS, right? So SDS will bind to one charge for two amino acids, all right? So with that charge, having negative charge, the protein will move toward cathode, which is the positive plus, right? So the apparatus that we use is something like that you see on your right side, right? So you have a buffer there, and then you put your samples in a land, and then your protein will move according to your to the size of the proteins, right? Small proteins will run faster, large protein will move slower, right? Okay. Good questions there, right? Now, let's say that we have our protein just now in our cells. Let's say the size is based on the color, right? Regardless the structure that you see there, right? Blue one is the biggest or the largest size, which is high in molecular weight, and the green one is the smallest proteins. Now you can see that by using SDS page, your protein can be separated based on the sizes. All right. So after you did your SDS page, of course, you can't see your proteins. You need to see them now, how they look like. All right. So that is detection, or what's called visualizations. You can basically stain them like you stain your clothes. All right. What we call as colorimetrics method. You use like Kumesi brilliant blue, which will give your proteins blue color, or you can use silver stainings. If you remember, well, you guys are all millennials. In the olden days, where people develop their photos, right, from negative, right, into photos, right, they use the same technique, which is silver stain, that you can see your proteins in silver color, right? Or you can use fluorescence. Which one is good? Number one, depend on your cost, all right? Fluorescence is expensive, but very sensitive. Colorimetric is cheaper, all right? But sometimes it's quite hassle because you need to stain and sometimes to destain to see your proteins. All right. So now, after you detect your proteins, you can see proteins in something like on your right side. It becomes like what we call as bands, right? And we use an apparatus called densitometer to scan. It's exactly the same thing like your scanner, right? But it's just that they have some function that the machine can basically quantify the density, all right? All right. If you see at the photos there, not only SDS page will give you the locations or the positions of the proteins, but if you look carefully, I purposely make it like you have like gradients. You see that the green like Pac-Man down there, the green Pac-Man is the lowest one. It's only in terms of quantity, it's just one. So you can see that after SDS page, it is just very faint band. But something like plenty, like the one like Pentagon in orange color, you have 2, 4, 5, 6, 7, I think 12 or 14 of them, means that the quantity is higher compared to others. You can see that bands is very dense. All right. So what you can get from SDS page is numbers of proteins, all right, their weight, all right, and also the quantity of proteins, right? Okay. Now, let's say that we by doing this, what we call as basic profilings, right? So we know now that 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. You have 12 bands. So you assume that you have 12 proteins inside, right? Okay. Now you can profile. How about doing some differential experiments? Let's say you have two same set of cells. One is healthy and one is sick. All right. Now this where proteomics is become very powerful to compare two different states. All right. So you want to know what makes the other cells sick, or when cells sick, what are the proteins involved? We would like to know that. So we isolate the proteins from both cells and we separate based on using SDS page, right? Now you have two different patterns from two different states of cells, right? So you can see that where in a 6 cells, this is just a make-believe, right? You have, can I show anything here? All right, can show. You have here, right, new proteins, right? This thing also, the position is a bit higher, right? these bands become very dense here compared to healthy states. In healthy states, these bands here, that's the density. Now, when it's 6, these become very dark, right? And you found that little green Pac-Man protein there gone in 6 cells, all right? So this is what we call as very basic differential proteomics using one dimension as the space. Why one dimension? Because we only separate by using one characteristics of the protein, which is the size of the proteins. All right. Can I stop here first? Right. Look at the slide carefully. What do you think about it? All right. Is anyone still there? Am I still online?
Yes, you are.
All right. Okay. I would like to, I put some questions there. All right. Hold on. Huh? If I share back again, sorry. I would like the attendees or the participant to see carefully. All right. Look at this. Anything that you can notice about these techniques in terms of look at the type of the proteins and the bands that they can detect. Anybody can point out anything that interesting, or you find that peculiar, or you find that strange?
Anyone have any comments on this simulated page? You draw by yourself, right?
I draw by myself.
Okay. A lot of effort. Okay. Students, anyone? Do you see any interesting things here? I mean, the things that weird or some things that maybe you want to
Anybody
know?
can point? If we are face to face, I definitely will give this like $1 if anybody can answer.
Okay, $1. $1
Well,
Zimbabwe.
you see that? Well, any takers? Who
Okay,
can
no,
point?
I got no response from the students.
All right,
Okay.
all right. If you see here, all right, in one band, it can be more than one proteins, right? Because the separation is just based on the size. Okay, many proteins have same size, so you will only can see them in as only one band, right? So if you look at the pentagon here, right, and then you see that this band become very intense or highly expressed, right? In condition B, we might think that, oh, this is one proteins, right? And the proteins become very intense ataupun very highly expressed during when they're sick, but not necessarily, right? Of course, we do not know this, but if you see that there's a new protein coming out, we don't see this stars in the first condition, in the healthy condition, but because they have the same size and they reside in the same position in the SDS page with the orange pentagon, we might think that, oh, the same proteins become expressed highly, but not necessarily. So this is the one of the disadvantage or limitation with SDS page is the resolutions that one band could be more than one proteins, right? So we probably cannot get the real pictures what actually happens, right? but it is still a very good technique, right? If you just want to start a proteomics work, right? Okay, all right. So let's do that. Well, let's say that. So now we have a technique that which is what we call as low in resolution. So what should we do, right? Right. Okay. So we have mentioned about two-dimensional electrophoresis. One dimension means that we only separate based on one characteristic. So if we can separate our proteins based on other characteristics, I give examples that in the class, right, you want to count, let's say you want to count boys and girls, right? That characteristic, gender characteristic, we can use to separate, right? We can use other characteristics as well. For examples, where they come from, right? Do they wear spectacles, or those kind of characteristics? Also, similarly, we can use for proteins. So in two-dimensional electrophoresis, number one, we separate based on size, and we have another characteristic, what we call as isoelectric point. All right, isoelectric point is the pH of a proteins, whereby, right, proteins have net charge. Some of them are positively charged, some of them are negatively charged. But at certain pH, the charge will become zero, no charge. All right, if they have charge, if we do electrophoresis, they will move, right? So if we have a gradient, what you can see here, it's like a gel with different pH. And we should what you see here is pH 4 until 10, right? And all the proteins have charge, right? If you put electric currents, so they will move based on the charges. If they are positive, they will move into negative. If they are negative, they will move into positive, right? While they're moving, the pH they pass will change, right? And the charge will become zero, and they will just stay put there, like they just park their car there, right? No more fuel, right? Charge is like fuel, so no more fuel, and that pH is what we call as isoelectric point for that proteins. So this is what we call as isoelectric focusing, or IEF. What you see on your right side of our slides is the apparatus, right? I use this kind of apparatus since I was undergraduate. Until now, this is the same apparatus that been used anyway. We only have two variants. This is one from Amasham, now is GE, and the other one is from Biored, right? So you can see that how high voltage been used to move proteins. 8,000 volt. How much is the voltage for in Indonesia? Is it 240?
120. 220.
120. Oh, 120 is late. Oh, okay. It's like
220.
oh, it's like Japan.
Hmm.
120, you don't die, right? In Malaysia, 240, you will die. Can you imagine if you get 8,000 volt? So the machine is like generating voltage, and you can see that how hard to move proteins, right? You need very high voltage to move proteins. Now, we usually will separate proteins first by the isoelectric point, so they will gather based on the isoelectric point. And secondly, we can use that, we get the same sample, we can run our SDS page. So they've been separated based on isoelectric point. Now they are separated based on their sizes. What you will see is something like this. So now protein with the same size but different isoelectric point, you can see them clearly. So now we have a better resolutions, and we can see them, and now we can analyze them if there are any differences, right? So this is what we call as differential proteomic using two-dimensional electrophoresis. First dimension is by using isoelectric point. Second dimension is by using molecular weight. Of course, you don't see something like this. That will be very helpful if you can see our protein like that. What you see is just circles, and based on what stain that you use. If you use CBD, you will see bluish. If you use silver stain, it will be something like very dark and brownish. Right? If you use fluorescence, you need a different densitometer where it can detect fluorescence. Right? So now it's your work to see. If you remember, I don't know. I don't think my son is doing that kind of activities. Let's say is that you have two same photos and you try to find what are the difference between the photos. Do you do that when you kid, right? So that is basically what we do in proteomics. So now you have condition A, that is the protein patterns, and condition B, that's the protein pattern. Now we need to see what are the difference, right? So we have a software, dedicated software that can help us with that, right? If your proteomes patterns is very simple, you can just immediately detect by your naked eyes. But if it's very complex, you definitely will need a software, right? So something like this, you will find that, you see that protein C, right, in condition A was expressed highly, but in condition B, the expressions become decreased. Now you see there's another new protein, G, just next to C, right? As well as protein A in condition A disappear in condition B, all right? All right. And same as others, as you will see that the protein D now disappear in condition B. So this is what you do in proteomics. So now what your interest is that what are these proteins and try to study what are the functions and try to relate what are their effect to the conditions that they've been expressed. All right? Okay. All right. So this gel base, all right, one of the limitations in gel-based proteomics is that you will see variations, because in science, you know that when you do experiment, you would like to minimize your variations. You do one time, you see different type of, you will see a certain patterns. You do second time, you see different patterns, all right? This is what we call variation. And these variations is what we call is one of the limitation in any SDS page, right? Because of the preparation of the buffer, preparation of the gel that you do. That's why some people, if they have lots of money, they can just buy the precast gel, right? They don't do it by themselves, right? So to eliminate that, eliminate the using of many gels, which will cause variations in your experiments. So people are very smart. So what they do is by using a concept what we call as 2D differential in gel electrophoresis. Means that in one gel, you put two of your samples with different conditions. So what you do, let's say you have protein from condition A and another one protein from condition B. Previously, what we do, we did two gels, but now you tag them with different color for fluorescence, right? For example, for proteins from A, you tag with Psi5, that's the chemical's name. It will give the blue color. And then another one is Psi2, where I give you yellow color. And then you pull these samples together and you run in one SDS page. Of course, IEF and SDS page. So you will get two samples in one pictures. And you can see that if it's overlapping, for example, blue and green, blue and yellow, if both samples have the same proteins, it will just overlap, right? Blue and yellow will give green color, right? Right. So you can see that immediately, right? For example, on your right side, on the right side of the gel, you can see that clearly there is a blue spot and a yellow spot there. So immediately you can point, that's the different proteins that from different conditions, right? Same as well on the bottom of the photos, you see that yellow spots coming out. This is very special. That only comes from condition B, right? Pretty nifty, right? It's just that this method is expensive because the chemicals is expensive, right? And yet you need a dedicated densitometers that can read fluorescence, right? The cheapest one still be the colorimetric staining method, right? Okay, you can use densitometer. When I was as undergraduate students, we didn't have densitometer, so we did use a normal scanner, right? The problem is just that because the gel is wet, so the water sometimes goes into the scanner and it's kind of broken, right? Those were the days. All right. So before we move on, so these are the apparatus that we use. What you see here is the large scale SDS patch apparatus because you applied voltage. When you run SDS patch, it will produce heat. Heat is not good for gels because it'll distort the gel, right? So you need, because it's big, so large heat will produce, so you need a cooler there. What you see on the left side, that is where the cool water will flow, will circulate to ensure that your SDS page will run as cool as possible, right? All right, so that is the example of a gel. This is really large gel, 24 times 20 cm, I guess, right? Very nice spots you can see there, right? And this is some of the apparatus that you need for scanning. Right? And the left side is the densitometers, where you can just pull the hash and put your gel inside there, right? It scans and you analyze with computers. You use a dedicated software that can help you with detecting the proteins. All right. Can I stop here first? All right. I talk too much. All right. Anything from anybody that can would like to give some questions or comments?
Anyone would like to ask any questions?
I hope that it's not hard.
They are trying to digest now.
Trying to digest. All right. Okay. Uh-oh. I think
okay, I haven't received the question so far.
So we continue, is it?
Okay. Let's just continue.
Oh, all right. Testing. All right, so now you have already separated your proteins. That now we want to identify what proteins, let's say you found a proteins that very specific proteins that only come out in certain situations, that you want to know what the protein is, right? So the strategy is for you to take out the protein spots from the gel, and then we analyze by using mass spectrometry, right? And after that, the data from mass spectrometry will be used for database searchings, and you will get your proteins, right? So that's a strategy. So mass spectrometry is basically a machine that will calculate or measure the mass of a molecules, right? But it must be in ions, right? Why ions? Because it's easy to handle. If molecule ions, you can just manipulate them. You can make them fly. You can make them circling things, right? So easier to handle, right? So remember, mass spectrometry is just a way to measure weight, but mass, right? Okay. So this is a basic component of mass spectrometry. You have ion source where you will turn your samples into ions. Okay. You have your analyzers, and then you have your detectors that will detect your samples, right? At least probably you have heard about MALDI right? ESI, those are ion source. Probably you heard about quadrupole, time of flight, okay? Fourier transform, those are analyzers for MS, right? So what's the difference between the ion source? That's how you ionize your proteins, right? Either you ionize based from solids or you ionize in liquids, right? Both techniques have advantage and disadvantage. MALDI is really fast, right? You put your samples and then you push your buttons. Within, I think, 2-3 minutes, if your proteins is clean, you can already determine your proteins, right? But electrospray ionizations technique will take some time, but it will give you a better coverage of what you analyze, right? So those are the machines, all right? MALDI, TOF, time of flights. Because the ion needs to fly, you usually sees the apparatus is kind of having like, it's like a lot of funnels, right? Right, those are MALDI, and then this is orbit trap. It's one of type of ion trap that, well, this was like 10 years ago stuff. Now they have a new one, all right? But this is one of top of the lines for mass spectrometry, right? This is just show you where the samples you put in MALDI. You put your samples in a steel plate, and then you plus your sample with lasers like Star Wars, right? For electrospray ionization, it will just have like very tiny holes there where your sample will go through, right? And then it will spray inside your analyzers. Mm-hmm. So this is strategy for protein identifications. Number one, you pick out your proteins from your gels, and then you do gel digestions by using trypsins. So probably you will ask why you just can't analyze an intact protein. It's too big for mass to analyze, right? And you need some patterns, right? So we digest our proteins by using protease. So now you know that different amino acids are composed with different sequence of amino acids. So if you break them, right, like trypsins, it will degrade at the arginine and lysine residues. So they will have different patterns if we have different proteins, right? So these peptides will go into MS, and what you get is a spectra of your digest, right? So now you have your spectra of your digest. By using such software, the software will help you in looking at the database, right, publicly available, and try to find what are the proteins that will have about the same pattern with your proteins, right? So if you have match, right, so that will be your proteins, right? And you already identify your proteins, right? So where did you get this database? That database is basically from genome sequences or protein sequences that people put in a public sites, right? Like gene banks, right? So sometimes this is what we call as peptide mass fingerprinting, but sometimes some proteins is difficult to be identified in such a way. Another way is by adding more information, right? By selecting the parent precursor ion here, right? You blast them further, you fragment them further, right? And you have more detailed spectra from that particular peptides, and you use that to try to find similar patterns of proteins in a database. By using double MS, we will call as MS because you fragment your proteins 2 times, right? You basically can read the sequence of your amino acids, right? If you can read the sequence of your amino acids, you can do homology search. Oh, all right. Okay. So those are the techniques, right? So you get your spectra. Basically, what you do, you have your spectra with their weight. You just kind of Google them by using something like Google. You put all those informations and you can already determine for something like this. Yes, your protein is probable transcription factor, something like that, right? So now the proteomics are accelerated by genome sequence, mass spectrometry, and bioinformatic tools. If you have a mixture of peptides, you can basically just analyze them in one shot, right? A mixture of many proteins, right? You digest them together, and then you run by MS, you separate them by mass spectrometry, and the power of bioinformatic tools and computer nowadays can just help you to determine that fragments belong to which proteins, right? That makes our life pretty easier, right? Not like SDS page, right? That we get one protein at a time, but if you have many protein, you can just blast them with by using MS, right? So this is the basic for gel-free proteomics, right? So rather than using gel, you isolate your proteins, right? You digest them together, right? And then you analyze by using mass spectrometry, and you identify the proteins that reside in your samples. In this case, I put here, you can do some such labeling, something like DIGE that I just showed just now. This is what we call as I-track labeling. It's just a chemical, one type of chemical that you can label your proteins, right? So you label proteins, different label with from different samples. You mix them. You separate them by using HPLCs, and then you analyze by using MS. You get spectra. Now you can see there your relative quantifications that can show you which proteins are higher. For examples, that orange one is higher than compared to the blue one. So this peptide means that proteins express higher in proteome B compared to the proteome A, right? So you don't have to use gel anymore. That makes our life pretty much easy, right? So what's the difference between gel-based and gel-free, right? Gel-based is quite hassles. You have to prepare your gels, right? You have to stain, and then it's pretty much like so many things to prepare. But what you get is an intact proteins where you can do many analysis because you have the whole things. You can do analysis, many analysis what you want. But gel-free, right? You destroy the peptides. Means that you degrade the peptide. It's a jumble of many things, and then of course it's pretty easy, but you need a good mass spectrometer, and you need to have a good software that should help you in detecting your proteins or peptides, right? Gel-based is also cheap, right? And then gel-free is a bit expensive, right? Nevertheless, right, both gel-free and gel-based are unique. They have, in these examples, if you use different types of method or techniques, you still get proteins that would not be covered in another techniques, right? So if you want to understand the whole proteomes, there will be never like one method can cover them all. Like Lord of the Ring, right? One ring cover them all. There's no such thing. You have to use many approaches for you to understand the whole stories. All right? Okay. I stop there. All right? That's nice, probably. Any questions?
I received one question, sir, but I'm not sure
Right.
whether we go now or after you completed your talk. I think over.
We can do questions, yeah. Or
All right. Okay. I just go
right.
for one question, sir.
All right.
The cost. Someone asked the student costing between gel and gel free.
Mm-hmm.
Which one is worth it?
Worth it. Oh, worth it. If you want cheaper, then it's gel based. Free, right? But as I said that, I didn't cover that. It depends on what proteins that you want to see. In gel base, many of these hydrophobic proteins is quite difficult to go into the gel, right? So if you notice, if you read paper journals using gel base, many of the findings, if you don't do fractionations, you will find that many proteins come from cytoplasm, right? It's quite difficult to find something that like membrane integrated one, right? But if you're using gel free, you can find such proteins.
Okay. I have more questions to come actually.
All right.
But I think I will deliver to you after you finish your talk first, Pak.
All right.
Because of the
Okay.
time. All right. Okay.
Okay. Let me speed up. All right. I can omit the cocoa proteomics. Let me know. All right. I can omit the plants part. All right. Okay, let's see some application here. You already understand how proteomics have been done. So can these tools be used in agricultural research? Yes, it is. Let's see. Right. This is a very basic one. The paper talking about they want to see if is there any difference in proteins between two types of cattle, right? One cattle which is have high RFI. I'm sure many of them, our students are from livestock, right? That means that less efficient in terms of feeding. You give them feed a lot, but they don't grow, right? And low RFI. If we can find some difference in terms of proteins. So they did two-dimensional electrophoresis and they determined the protein based on ESIMS, right? So this is the basic that we talk about. And you see that paper is 2018, just 2 years ago. Of course, it's been done like maybe 4 or 5 years ago, all right? So they separate based on SDS page or 2D page, and they identify some proteins. So they see that in less efficient kettles, they found that this protein, hemoglobin subunit beta, is highly expressed in a high RFI kettles, right? So what does it mean then? We covered that later. Another one is the heat shock proteins, also highly expressed in less efficient kettles, all right? In a very efficient means that low RFI kettles, three sets of proteins, fatty acid binding proteins, all right, and malate dehydrogenase, which are the mitochondrial, which involve in mitochondrial functions and energy metabolism, are expressed highly, right? So what does it mean here? They infer that because this hemoglobin subunit beta is related on oxygen transport and blood flow. So before them, they found that the cattle that having high RFI has very high in terms of hemoglobins, means that they use lots of energies, right? So that's why in this paper they found that the proteins also highly expressed, right? And then the protein of heat shock proteins is involved in energy expansion, means that if they have difference in energy expenditure, means that that will involve in the efficiency of feedings, right? And finally, we have this mitochondrial function and energy metabolism proteins that is very highly expressed and in low RFI, which means that high efficient kettles Now we know that these proteins related to energy as well, means that if there is any difference in terms of how these cattle use energy based on their feedings, of course there will be if they can efficiently use the energy, of course they will eat. So the efficiency of the feeding also will be better, right? So by findings of this work that they found that there is differences in terms of proteins between high RFI and low RFI in beef cattle. All right. So what they can use here, the proteins, if they can use in breeding program for selection probably, right? They could find that proteins that cattle having high expression of hemoglobin subunit beta will be having high RFI, so they can screen out when they do breedings. All right. So that is one of the applications that people use in cattle research, right? So the second application here that I would like to share is people looking into try to find differences or uniqueness in their products, right? Here they have this research is from India. When this researcher are looking into the constituent of milk in cattle, right? Two types, Kashmiri and Jersey. So Kashmiri is the Indian origin cattle, right? So they try to find what is special between these two cattle in their milk. So what they do is by using gel-free proteomics, and they found several proteins that are expressed differently in the two breeds. So the most call this the most interesting one here are the FMO3, which is flavin containing monooxygenase 3, which were found really highly expressed in Kashmiri cattle. Now, I would like to say, I would like to apologize. I put 17-fold under drug metabolizing enzyme. No, this 17 folds is FMO3. Among all the proteins, FMO3 was found very highly expressed, 70-fold compared to just C ketones, right? So what is the proteins do? After they did studies on some functions of the proteins, they found that this FMO3 is oxidizing trimethylamine into trimethylamine and oxides, right? So trimethylamines will give these compounds have fishy odor, but FMO3 will make into odorless. So that will make the difference between the taste of Kashmiri cattle and Jersey cattle, right? Not only that, they also found that proteins involved in immunity, CD4 antigen and lactoferrins, and as well as drug metabolizing enzymes, right, are found highly in Kashmiri cattle. So they're thinking about this Kashmiri cattle could be a functional food, and probably can be given to the probably baby, right? So these findings actually can help them in promoting, and probably farmers can get better price for Kashmiri cattle compared to Jersey cattle. And we can see here as well that proteomics can be used to differentiate two different cattle breeds. All right. Okay. This is another interesting work talking about chickens, right? Well, in right, here's the rationale of study first, right? So, well, egg hatchability is very important parameters for reproduction performance. Even though the same hen, okay, having same age, same breed, and then you put in the same environments, and the egg that they produce having same quality and they incubated in same environment, but many can see that the hatchability of eggs are different, some having high hatchability and some having low hatchabilities, regardless the similarity that they have, right? So this must be coming from somewhere from the genetic of the hen itself, right? So the researchers are looking into the in the white part of the egg, right? Many people think that the white of the eggs is only supplying water for the embryo, but actually inside of the white egg, there are pretty much of proteins that are very crucial in terms of development of embryo, right? So they would like to know if they have different type of different set of chickens having different hatchability in terms of the eggs, right? Can they find markers that can differentiate between high hatchability and low hatchability eggs, right? So that is what they want, right? So they did the proteomic analysis. They have two different sets of chicken that surely can have high hatchability egg and low hatchability eggs, right? So they did their proteomic analysis by using isobaric tag. All right, this isobaric tag, something like the eye track, means that you have two different samples, you tag with different labels, you pull them together, and then you plus it with LC-MS, and then you depends on the software to give you the results. All right. So what they found, all right, so very interesting here that they found that in a very high hatchability egg, they found that proteins with neurodevelopment that involve in neurodevelopments, antimicrobial activities, right, such as mucins, and neurodevelopment in keratins is really highly expressed in high hatchability egg, right? But in low hatchability egg, they found that the proteins that involve in blood coagulations are low. So they think that by using these proteins that can differentiate between the two conditions, they can use them as a biomarkers to predict whether the chickens can have high hatchability or low hatchabilities. Right? That will be very beneficial for the poultry industries. All right. So before we end, right, there's another one talking about mastitis. We have this Streptococcus xylosis, which is one of the bacteria that cause mastitis, right? And we have problem because they have if we use right regularly, that probably the resistance will develop, right? So what they did, they used antibiotic tylosins to treat it, means that they treat the bacteria and they compare with untreated, and they use eye track here, which is a gel-free LC-MS method, right? So they found that proteins in tylosin treated proteomes, translation-related proteins are really highly expressed, right? So proteins that highly expressed during those times, they infer that they could be involved in the mechanism of resistance. So this protein can be target for any antibiotic or drugs that will be developed later on, right? All right. So,
Sorry to
yep.
interrupt. We have only a few minutes left before.
Okay, so that's all I think. The application, the others are my works because this is our plants. So we did some work on cocoa as well to see any difference between two types of cocoa, all right? Tolerant with pests and non-tolerant with pests. We did find some interesting proteins, right? Like polyphenol oxidase that involve in defense in tolerant varieties. We found trypsin inhibitors that also intolerance, all right? Chitinase-like protein. These are all proteins that may involve in the resistant or tolerance characteristic in cocoa. So this is what I did, all right? So here's the thing. So as to summarize, we know that proteomics is a study of the whole proteins, right? And we have two types of approach, which is gel-based or gel-free. And if we combine with mass spectrometry and bioinformatics, it can be powerful ways to study the proteomes, which is very complicated. And by sharing few applications, right? Just now that we know that proteomics can be a useful tools in agriculture research to address well many agriculture issues that are now currently that we have, right? With that, I will say thank you very much. Really, thank you for listening. All right.
Thank you very much for the presentations, Dr. Azwan. It's a very interesting lecture, and I received actually some questions already for the students. So some students are in queue to ask. I don't know whether we have enough time to accommodate all or no. I will go for the first student, Anissa. Are you there? Nur Anissa.
Hello, Anissa.
Hello, sir.
Okay.
Hello.
Proceed, Anissa.
Yes, I'm here.
Please make it brief. We don't have so much time, so
I'm
make it brief
sorry,
so that
maybe
the student have time to ask also.
Oh, okay. Sorry, sir. I come back to the middle slide about the detection of a sick cell based on proteomic differences. You said that a sick cell and the normal cell has the differences on proteomic characteristic. We
Right.
can analyze it with SDSPH or gel electrophoresis.
Yes,
Can we detect the specific problem on a cell based on its analysis? I mean, we just know there were a problem, but we don't know what is it, or we can know there are some problem on RNA or on cell membrane specifically.
all right. All right.
Thank
I do
you.
understand. All right. All right. Very good questions. All right. Now that you have two types of situations, you always want to see, let's say you have a healthy one and a sick one, and then you want to know why the cells become sick or something else, right? So that will be depends on the approach that we'll use. You said that you ask either we can find what the culprit or what caused the cell sick, right? Because we only look at proteins means that we can discuss only on proteins and depend on the set of proteomes that you managed to cover, right? And proteomics will provide you until the determination of proteins and you try to infer the functions, right? You have to follow up by, like, for examples, experiments like something like, I don't know, like knockout experiments to see whether the genes is really, involved in that particular mechanisms, right? But until for proteomics, you can know the proteins and you try to infer the functions, right? Based on the such as there's a different type of software that they can use to try to understand the function of the proteins. Am I answering the questions?
Okay, thank you, sir.
Thank you, Anissa.
Thank you very much.
I will move to next students. Sabrina. Sabrina, is it correct? Are you there? Is your microphone on?
Hi, Sabrina.
Can you hear me clearly?
Yes, I can hear you clearly.
So, sir, hello, my name is Sabrina. I'm from
Yes.
IPB University. So my
Yeah.
question is, what kind of research you usually do, or what do you want to know usually by separating protein? And is there any difficulty when you did the experiment?
All right. Thank you very much. All right.
So,
Very good questions. All right. I didn't have time to share my work. I'm working with cocoa, so we want to see last time what makes certain cocoa variety very tolerant to a pest, right? Which is cocoa pod borer, which also Indonesia have the same problems. So we found that certain cocoa varieties are tolerant, and we want to know what make it tolerant. So we want to see at protein levels what make them so, right? And by doing this, we really hope that we can use that specific proteins probably as a markers when we do breedings to help us screens. And currently, what we are doing is also we have projects on trying to understand what makes cocoa flavor different, which is due to fermentations. And cocoa flavor comes from proteins, right? And degradation of protein during fermentations is very important. So that is what we are doing currently. And we also try to see what are the proteins that probably having interactions. I mean, that livestock, right? The current issues now we have is COVID-19, all right? So we want to know if the virus protein do have interactions with proteins from livestock, which later on probably we can understand whether livestock also susceptible to the virus, right? So that kind of thing. So what are usually the problems that we are facing for gel bears? For plants, we have problems with the preparations of proteins, because plants, most of plants that I handle, what we call as tough plant, like cocoa, right? Cocoa have lots of polyphenols. They also have lots of gums. So trying to make the samples pure means that try to remove all these contaminants is one of the hurdles that what I had during the work, right? And last time, 10 years ago, when we have not have DNA sequence from cocoa, we have to sequence the amino acids. Nowadays, determination is based on, if we have DNA sequence, you have database to find your proteins, right? Those days we don't have that. So those are among the hurdles that we have.
Okay, I hope it answered the questions. Sabrina?
Did
Okay,
I answer?
Yes.
We have
Thank you, sir.
But I hope by sharing the difficulties in the experiment, the student won't afraid with proteomic, yeah?
Yeah, you have, that one thing as well, that proteins you cannot amplify like PCR, right?
All right,
so
okay.
if you have
We
small
still
proteins,
have we still have many questions to come, but I think unfortunately due to time, I will give only one chance to not the last questions, maybe for Nur Hafizah Muhammad from University Putra Malaysia. Hafizah, can you turn on your mic?
Yeah, she wrote something here as well.
Yes.
Can we predict?
All right, okay.
Yeah.
Proceed, Hafizah.
Hi, I'm Naraviza from UPM. So my question is can we predict the proteome dynamics using the gene expression data that we already have?
Gene expression data.
Yeah, for example, from the qPCR, we will obtain the gene expression. So can we estimate the proteome dynamics? For example, the abundance of the proteins data.
All right. Yes, you can, but because we're talking about products of DNA, right? Means that product of DNA can be either transcriptome or proteomics. If you have data from transcriptomics, you can guess the proteomics behavior. But remember that transcriptomics, the transcriptomes, not necessarily correlated with proteomes. That's why discussion only by how I can make it, like you try to infer the proteome dynamics based on transcriptomes may have some what we call as limitations. All right. Yeah, you have to look at the protein levels, right?
All right. Okay, Doctor. Thank you so much.
Yeah, sure, no problem.
Okay, I think that's clear enough. But maybe if I may add on also the proteomics still can complete the data from gene expressions, because sometimes the RNA expressions doesn't completely reflect the protein level, because the RNA may degrade somewhere and then it won't be completely converted into proteins.
Yeah.
Okay. Anyway, why I have to answer this?
Because you like it.
All right. Okay. I think because of the time, I think actually there are several questions left on my notes. Yeah, the positions of proteomics and so on. So why we need to digest with trypsins? Will it because it will destroy the protein and so on? I think some students raise the questions more on technical things.
Oh, that's good.
I will deliver to you after this. I mean,
yeah, sure, I can answer.
You may answer it in written form later, and then we can send to the students.
I will
all right. By this, I think I would like to end the sessions. But before that, let me share this token appreciations from the committee for Dr. Azwan. If everyone can see my screen.
Yeah, I can see. Right.
Okay. So since everything is online, so the handover of the certificate of appreciation also on screen.
Oh, sure.
By email. So this is a certificate appreciation for you for delivering a lecture on proteomics in agriculture in the program.
Can I take photo of that?
Sure.
All right. Okay. All right.
All right. From the Department of Animal Product and Technology, Prof. Irma, and also thank from the Dean of Faculty of Animal Science. Hope you can visit again sometime to Sandakan, seafood in Sandakan.
All right.
Thank you, Pak. I think that's all. I would like to end up the session. How about the screenshot for documentation?
Thank you very much. Yeah, thank you.
Nice
Thank you very much. Probably it was too detailed.
Okay, sorry. I think we have some feedback voice from students. Can all turn off the microphone, please? All right. Anyway, so do we need to take? Do we still need to have a screenshot for documentations, Bu Tuti?
No. Who, me?
No. We sometimes need the documentation. Anyone please turn on the video, and then the committee will take a picture for all the screen for the documentation's purposes. Or it's done already. Otherwise, I will just stop it here. Sudah, Bu. So I'll see you again in the other occasions. I think for sure the committee will have will invite you in the other occasions after this. Thank you very much. We will move to another section. Thank you. And of course, if you want to keep joining us, we'll be happy for us. But if you have something else to do, then you freely leave the platform.
All right. Thank you very much.
All right. Thank you, Dr. Azwan. Thank you. Bye-bye.
Okay.
Bye.
All right.
All right. Okay. Let's move to the next session for the. Did I still sharing my screen? Yes, I do. All right. Okay. We move to the next sessions for. I cannot see this one. Okay. Okay, we move to the next session for the virtual excursion session by DB Food. We have already Pak Heri Kurniadi, Sarjana Peternakan. Pak Heri, are you there? Halo Pak Heri.
Ya, halo, bisa dengar suara saya?
Okay, nice to see you Pak.
Okay.
I think I leave the stage to you for presenting for give us some virtual view on your company. All right.
Okay.
The time is yours, Pak.
Okay, thank you. I think I get some troubleshooting here.
Okay. You want to say any video or something?
Yes, I have a PowerPoint, but I cannot access. Wait. I have a bad connection here.
Pak Heri, you want to share any
Yeah,
video?
yeah.
Or maybe you can
I guess I have some troubleshooting here.
All right, okay. All right, while Pak Heri is troubleshooting the things, Prof. Asep informed me regarding asked me to remind the student regarding the assignment for the review. So as Pak Asep already explained about the assignment for the review yesterday. So do bear in your mind that all students can choose any topics that you are interested as a topic for your review, and then you write down in the essay format as explained yesterday. I think the format already recirculated to the students, and also that review for those who are interested. Also, you can use review to present during ISC, International Student Conference, sometime on September. I think I don't know, maybe third week of September or something. I think detailed announcement will be done. So also for those, let's say, would like to took up the proteomic topics for as a topic for your review, feel free to go. Just write down whatever you got from the previous lecture presentations, and also of course you can add on more from the papers that you read by your own. I think.
Okay.
All right. Okay. Silakan Pak Heri.
Is it now on screen?
Oh, freeze.
You get my screen?
No, not yet, Pak. Okay. Okay. We got it. Yes.
Okay. Thank you. Good afternoon, ladies and gentlemen. Thank you for the opportunity you give to share a little of my experience in starting a business based on livestock-based industry. Our company using the brand DB Foods and have a company PT Banteng Perkasa International. This is our product, the first product, but also the backbone of our product. We call it dendeng balado. It's a traditional culinary from West Sumatra, Indonesia. And this is also rendang. I think everyone in this world know about rendang. It becomes more popular after the CNN took rendang as the first delicious food in the world. And here's all of our product that we produce until today. Now, this is the history of DB Foods that I want to share to all of you to give a perspective how to start a business, a livestock-based industry. The early history of DP Food is a bakery business that founded by IPB students, including me, in 2008 with initial capital of 2.5 million rupiah. In 2029, DP Foods was established in Padang without a single employee and focused on the processed meat business. This is a different part of the history, because when I graduated from IPB, my university, I see the bigger potential business based on meat than bakery. That's why, and that year, I get all my capital goes to my hometown in Padang and start this business. DP Food obtained a halal certificate from LPPOM MUI and became the first packaged Padang beef jerky products in Indonesia. In 12, DP Foods received a business contract worth 500 million rupiahs, and DP Foods products were sold at 50 hypermart outlet from Medan to Abepura. And in the same year, DP Food was chosen to represent Indonesia at the International Expo Food Taipei in Taiwan with Garuda Food and Sostro. We know Garuda Food and Sostro is one of the largest food company in Indonesia, and also popular in abroad. So at that year, that is a very monumental and a very good improvement for DP Food to side by side represent Indonesia in Taiwan with Garuda. In Jakarta, they have about 12 premium outlets in the premium mall, including Grand Indonesia, also in Pesanggerahan, West Jakarta. And we also into the competitor of French market, the food hall. Now, we also get our product into the Kemchik. Maybe some of students knows about Bob Sadino. Bob Sadino is one of the legendary that makes many students try to be a businessman with all the greatness and the passion that he teach to us. We also get in our product to Galail's signature. The Galail is the same owner with KFC, Kentucky Fried Chicken. They also have 2 premium supermarket in Jakarta and outside Jakarta. And this is also the market city. The premium supermarket in Pantai Indah Kapuk, North Jakarta. Maybe we know Pantai Indah Kapuk is one of the greatest and richest people in Jakarta, and they sell our product here. This is Lulu supermarket and GS supermarket. We also throw our product to this chain supermarket. Why we get our product into Uni Emirat Arab Lulu and South Korea GS? Because we think our product can be exported to the origin of their country if the people who buy this product in Indonesia in their supermarket have a good point, and that makes sense. Our product someday will sell in the origin of the country. Okay, where are we going today? We start our perspective with Indofood. This is the revenue. We know Indofood is the largest instant noodle company in Indonesia, also in the world. After this, we see Astra International, and then we see Smithfield Food. The Smithfield Food is a livestock-based industry in United States that we can see the revenue of Smithfield Food is much higher than Indofood in Indonesia. And we can also see Tyson Food. It's the same livestock-based industry in the United States. And the revenue is very, very higher than Indofood. If we see this 5.1 versus 30 million dollar US, it's about 7 times. It means a livestock company can make revenue 7 times than the largest instant noodle company in the world. That's the point that we must learn. Because many people think that a livestock company or livestock business is not a good business. But actually, in Europe, in United States, a livestock company can make a high demand and high revenue business. That's why we, as a student of animal husbandry or a livestock faculty, we must change our minds that we can make our business in livestock company can make revenue 7 times than Indofood. If we think like that, we can make a global change into our business in livestock. This is our vision, the most respected, fastest growth food company in Southeast Asia. And the future of DB Food, we talk about the future. The future of DB Food, I tell to our member, we call it Future 7. The first, consumer goods. Second, restaurant chain. After that, premium retail, livestock farming, spices plantation, biotechnology. This is the 6th. And the 7th is space food. The space food is the end of the line of our perspective. Because the space food is completely the biggest thing we ever see, that we can make a great food outside the world. Before go to the space food, of course, we must take the biotechnology business, because there's a lot of things we must think about before we get the space food company. So my father once said, as long as humans have not eaten stones, the food business will survive. That's why DB Foods will survive for the long time. Thank you.
Thank you, Aheri, for the descriptions of the DB Foods and also all the product from the companies. I believe that if you have any comments or questions to DB Food students, if you have any questions or comments, please you can ask directly. Just turn on your microphone. You have Hanif? Hanif, we cannot get your voice. Can you?
Okay, test.
Okay, all right, okay. Just proceed.
Good afternoon, Mr. Harry. Sorry for the loud. Okay, so my question is, let's say I have 100 chicken in my house. Like I try to produce a poultry production, but I only have 100 chicken. So how can I sell the chicken after apa namanya? It end of the production to sell it, but have high, let's say high, not sell it in the market. Like what I see is when we sell in the market or let's say pasar, yeah, it the price is very low, like 40,000 or maybe 30,000. But when we can, let's say, McDonald's can sell a burger for about 40,000. That just a little bit of part of the chicken or the meat. How can I start to have a big income from 100 chicken? Let's say like that. Thank you, Mr. Harry.
So you, let's say you want to supply your chicken to DB Food or the other company, something like that, right?
Yeah, how to have a big income from the little amount of chicken.
All right, okay. Please, Pak Heri, I think it's clear enough, the question.
Yeah. So it's the same question I get before I start this business when I already graduated from APB. Maybe some people know about marketing. Some of the thing about marketing is STP, segmenting, targeting, and positioning. So we talk about STP, the segmenting. We must divide the market into 4 or 5 segments, and then we target which segment that we want to go. In DB Foods, we targeting the segment, the highest segment, the premium segment, also the premium customer and premium supermarket. Because if we want to get more revenue, we must sell it into the premium customer, into the premium market. For example, one of our product we call rendang. One piece of rendang we sell in supermarket about 27,000 rupiah. Okay, one pieces rendang we sell 20,000 rupiah. But if we look at the traditional or modern restaurant, Padang restaurant, with 27,000 rupiah, we can get not only rendang, but also rice, vegetables, maybe some crackers, and many more. Maybe we can get some iced tea with 27,000 rupiah. But DB Food can sell only one piece rendang with 27 rupiah. Why? Because the people that we segmented is a premium customer. What's the key to get into the premium customer and premium segment? There's a lot of things. First, the quality. The premium customer need the best quality. We offering the best quality. The second, they also don't want preservative or we call it pengawet. Okay, in Indonesian we call pengawet. The premium customer don't want unhealthy food. And the third, premium customer need the space, the premium customer actually buy the premium food near their place. Okay, so we need to sell our product near the house of the premium customer. Place, we can say the key is the place where we sell the product. With a good place, the good quality of food, we can sell more to the premium customer. Okay.
Okay, thank you, Pak Heri. Any other questions from students? Hanif, I think Hanif, you really have the chicken in your home, right?
Yeah, I try to grow some chicken.
All right. Anything else, Hanif, or from the others?
Can I ask more, like, to get the best quality and healthy foods, like pengawet, let's say preservative component, we need to have a good standard. Like example, the kitchen, let's say the kitchen might be hygiene, but in our house, in my house, example, we just have a house ordinary kitchen. But how can we produce a high quality of food, like high hygiene, like we promoting the hygiene, the quality, and the best production of our poultry, let's say, to the consumer in this stage, in my stage, like that. Thank you, Mr.
Okay. I think you must rent a space. You rent a space, so you make a proportional kitchen in that space. House, rent a space, a little space maybe, but it can use as a kitchen. Rent a space, sewa lokasi untuk dijadikan. Okay, because the production for premium market, we need a proportional kitchen, not a house kitchen.
Okay. Thank you, Pak Heri. Okay, I think we still have a few more minutes. Maybe if one more questions from the others. If not, that if any more, one more questions.
Sorry, Pak Cahyo. This is Raisen Farida.
Oh, okay. I cannot see who raised the hand. Okay, siapa? Farida. Okay, please go ahead, Farida. Farida, are you there? Hello. Anyone else who raised the hand? I saw someone, but I don't remember the name.
I want to ask something, may I?
Okay, Adib. Proceed, Adib.
Yeah. I'm sorry for the video. I cannot show my face because my webcam is troubled.
Okay.
Hello, Mr. Heri. Good afternoon.
Yeah.
I'm Adib Zainuri from IPB. Want to ask something from your brand in rendang. In the logo before, it said that the rendang use tenderloin meat. Is that right?
Yes.
May you tell us what type of cattle is the meat come from, like Bali cattle or maybe Angus cattle? Thank you. That's my question.
All right.
Okay.
Thank you, Adib.
Actually, in Padang, we get the cattle from Lampung. All the cattle we use from Lampung Sometimes we use a Brahman cross, but sometimes we use maybe Brahman cross is 60% we use for rendang and dendeng. Some cattle maybe from Bali, but it's not a big number.
Pak Heri, just curious if the breed of cattle really affect the rendang quality?
Yeah,
Oh.
of course.
And
Because the simple is like that. If we use too old cattle, the quality is not good.
Okay.
But if we use too younger cattle, also not good. Okay. The very good quality of meat is in the middle egg. Okay.
All right. I think unfortunately I have to end these sessions because the time doesn't allow us to proceed. Thank you everyone for participating in the sessions. Thank you Pak Heri for the very good presentations and good overview about the DB food. I believe that the students are really interested to know more actually, but I think because of the time we cannot have more questions to convey. So before I end up, I would like to share the token appreciations from the department for the DB food. Let me share my screen. Okay, all right. Can everyone see my screen? Okay. Okay. This is a certificate of appreciation for DB Food, Pak Heri, on behalf of Department of Animal Product and Technology, Faculty of Animal Science. I would like to extend our sincere gratitude for the DB Food for the contributions on the program, on the summer course program of Animal Product Technology, and appreciation also on behalf of the head of department, Professor Dr. Irma, and the Dean of the Faculty of Animal Science, Professor Dr. Sumiati. Thank you very much again. Thank you again, Pak Heri. The file for the certificates will be sent to you by email. Thank you for supporting our program as well. Thank you very much. So,
thank you very much.
Okay, I would like to end this program, these sessions, and we move to the next sessions for the another excursion for the cultural heritage of Indonesia. But before that, let's give a big applause to Pak Heri, and thank you, Pak Heri, for being with us. And please feel free if you may leave at any time. All right, okay. Let's move to the next session for the excursion program for overview of Indonesia culture. For this, we have 15 minutes of overview. It's not 15 minutes, it's 10 minutes of overview. For today, we will go to Yogyakarta, one small province located a south part of the middle of Java. Am I correct? Okay, anyway. All right, okay. So I leave it to Pak Arman to the overview of the culture. Pak Arman. All right, okay. So please enjoy, everyone.
Jemas, no sound Jemas.
temples stretched far and wide as the legacy of historical greatness, like the ones found in this 15th century palace of Cirebon. Take the train, let it take you back in time, and enjoy the timeless feel of a magic ride. Here, every tale is expressed by genuine human relationship and by the deft touch and tender care of artistry. The great beauty of batik has a life of its own in this land. Time-honored values are preserved in body and soul. This intimacy is formed. In the capital, tradition comes in style. Urban festivals frequently grace the streets. Local dishes and international menus are presented. In the warmth and friendly manner Bandung is known for. Fashion and lifestyle have long been the synonym of the city. It is a shopping nirvana. The lush and beautiful landscape offers only the best of serenity. From the five-star hotel's luxury to the intimate lodge's tranquility. You can always take a journey to the mountain and walk down to the crater. It also has the most extensive botanical garden in the country. It's a perfect choice whenever you want a place of solitude. Your time and effort will be immensely rewarded by the spectacular beauty of West Java. Wonderful Indonesia.
A wondrous civilization. Disposed to tranquility. With scenic landscapes bordered by the sea. Here, mythologies become principle. And religions create the cornerstones of life. It is contained in the arts and crafts, where celestial ceremonies become part of popular culture. Legacies of glorious traditions, where folk tales are regarded as guidebooks to perfection, honed over centuries. yet possible to experience within a few hours. Experiences you can take home. Here, art preservation is a tourist attraction. History is a living monument. Even shopping is meaningful. See world-famous resorts compete with local means of accommodation. Enjoy natural kindness. Or the excitement of exploring nature. With endless stories to tell. And never-ending feasts. Never-ending possibilities to get more out of life and journeys. Yogyakarta. Wonderful. The mountainous region of East Java. Where Majapahit once stood. Largest ancient kingdom. Changing and opening slowly over hundreds of years. A historical city on the move. A celebration of remarkable heritage, grandeur of the past, wondrous ancient architecture, and the outstanding universal values behind, where art absorbs the audience into colorful drama. Still, the exotic mesmerizes. Extraordinary feasts exhilarate. And bring us back to the land of the ancient mountains. A myriad of outdoor adventures. In exceptional panoramic nature. Exciting variety. This is a land of historical significance, where the old days are celebrated. Local cuisine is cherished. Beyond the city, preparing for a thrilling ride on the vast sea of sand in Bromo, East Java. Wonderful Indonesia.
Okay. That's all. Pak Jimas, Pak Arman, finish? All right. Okay. So that's all for the overview of the Indonesian culture, particularly for Yogyakarta. Yeah. So I think by that I would like to end the sessions of the virtual cultural excursions. So then we're gonna move to the second lecture today, but I think we will have a break for supposedly start 3:45. So 10 minutes break before we start the lecture. Is that okay? Okay, yeah. So okay, let's have a break for a 10 minutes break, and then after that, everyone we can back to here, and we can start the sessions. All right. Okay. See you again, everyone, on 3:45. Okay. Hello everyone, we're going to start in 2 minutes. Please be ready with your screen. Okay, another 1 minute. Okay, we start. Let's start the next session. While waiting the second presenter, please turn on your video to display yourself so I can identify whether you all are here or still stuck in toilet or somewhere else. Okay, good. I can see Mariam Zuhdi, and then the rest still off. Farida Cynthia, still off. Sam Amatori, still off. off, and then Salma, Farah, also still off. There are many of you still off. Gabriel Buccelli is still off. Gabriel. Ismi still off. Okay. All right, I think everyone is back. Almost everyone is back, but I still see some videos still off. Some of you face still like just woke up from your sleep. All right, okay, let's see. The second screen I saw still many off. Farah, Sekar, Farida, Gila. I don't know, cannot see the names. Second screen, Asila, Raihani, Aliani, Sakira, Jin Suang, Anissa. Hello everyone, are you there? Hani Azmi, Akila. Okay. All right, many of you still haven't turned on your video, particularly for the students. Okay. Oh, I saw Devi Murtini. And I just realized also I didn't turn on my video. All right, okay. Okay, everyone are ready? I don't consider you are ready before you turn on your video, yeah? Students, I'm talking to students. Nurul Dini, Farah. Are you there, Farah? All right, Dian, just turn on. Okay, Aliani, Amin, Shakira. Oh, we have Shakira, we have a singer. Maulita Putri, Raihani Hasna. Akilah, Hani Azmi, and Carlina Yusfa and so on are still in. All right, okay. Oh, still many of you not on the screen yet. All right, anyway, okay, I think I don't want to delay more. Let's start. Bu Irma, ready? Okay. All right, okay. All right, welcome back everyone to the summer school program. We're gonna end up this today schedule by moving to the lecture 9, not lecture 10, sorry. Let me share my screen. All right, okay. I want to have everyone on the screen because I have a very interesting picture on my slide from Korea. I want to share to everyone. It's a beautiful picture. But if you are not ready yet, then right, okay. All right, okay. So we move to the lecture 10, which supposedly by Dr. Azwan, but we swept it already. We'll have a lecture from Professor Dr. Irma Isnavi Arif from IPB University. She will deliver her lecture on traditional animal product processing in a few minutes after this. Before this, I think Prof. Irma is ready for her presentation, but before she start the talk, let me introduce her. Okay. Professor Dr. Irma Isnavi Arif, traditional for the talk of the traditional animal product and processing. Okay, she's from Indonesia. She's just wearing traditional Korean clothes, so don't be confused about that. All right, Prof. Irma is, let me introduce her briefly. I have a long CV from her, but I just cut it some parts only because I don't think we have enough time to read all parts of her CV. Prof. Irma now is an active lecturer in divisions of animal product and technology, and is also a professor for this field Faculty of Animal Science, IPB University Indonesia. She graduated from Faculty of Animal Science in 1998 for her Bachelor of Science, and then earned his MSc in Food Science in IPB University, as well as her PhD in Food Science in IPB University in 2011. Along her PhD program, he also got opportunity for having sandwich program at Osaka University from 2019 actually to 2010 to 2009, 2010. I think it's right. Okay. All right. She joined IPB University as a lecturer since 1999, a year after she graduated. And then after that, she held several management positions in the university since 2003. She's one of the board management in IPB boarding houses, and then head of Animal Product Technology Divisions, faculty senate member, head of Animal Product Technology, and also an university senate member. Since 2015, she held a position as HOD, head of Department of Animal Product and Technology, until now. All right, 2 years more to go. Okay, she's a very productive researcher. I don't need to list up all her publications, but you can see her productivity from the Scopus with 8 H index and then citations more than 150, and then 12 H index under Google Scholar and Web of Science. Under Sinta is ranking in 119, which is considerably very good, very prolific researcher in Indonesia. The research experience of Professor Irma lies mostly on the food fermentation, animal product fermentations, spanning from milk, meat, and also some other natural products applications on the animal product development. She received several awards as Bedhoff HOD at IPB University level in 2017, and then best poster presentations during the research seminar held by Ministry of Research and Education of Indonesia in 2015. Also one of the Indonesian innovator in 2015. Yeah, I didn't put here how many patents he has. And then also best oral presenter under Indonesian Story Science Foundation seminar in 2014. And then civil service long service medals from Indonesian presidents in 2010. I think she also have, I think this is the last part. She also been trained in wide field of lab or research fields, spanning from chromatography, protein engineering, and then molecular genetics, and then also some training in nutraceutical and functional food, and genomic using domestic animal as genetic model, and so on. And recently, I think she went to Thailand for bioinformatics for metagenome analysis. All right, I think by this, I would like to deliver the stage to Prof. Irma to start the talk. But again, before she start the talk, I would just remind, I would like to remind all the students, please turn on your video as I requested earlier. And then second thing is please fill in the registration form, not registration form actually. The presence link, yeah, attendance link. Sorry, attendance links. Later, the committee will share the link on the chat room. You need to fill the attendance link twice, yeah, at the end of at the beginning of the talk and also at the end of the talk. And then lastly, if you have any questions, I would like to encourage you to ask directly to the lecturer, but please let us know first, yeah. So chat me privately or raise your hand so that I can call your names, and then we will turn on your microphone to start your questions to the speaker directly. All right, okay. And for those who has no problem with the webcam, with your camera, please turn on. Okay, this is mandatory. Okay, I think Prof. Irma, I think the time is yours to start your talk. Thank you.
Thank you, Dr. Cahya. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone, and also good evening from our college in Malaysia, also in Australia, maybe. Thank you very much for the committee to allow me for sharing our knowledge. Actually, this is our some presentation is based on our research with Dr. Cahyo. So maybe in the middle of presentation, maybe Pak Cahyo can give more detailed information about this. Thank you, Pak Cahyo. So I would like to share my screen. Okay, anyone can see my slide? Yes. Anyone can see my slide?
Okay, it's on screen.
Okay, it's okay.
Okay, today I would like to talk about the traditional animal product processing. Let me check. Okay. Why we would like to share about this because Indonesia has many animal products technology. So we can share how the beneficial, how the function of animal products based on from Indonesia. Okay, the outline. I would like to talk about the fermented food and non-fermented food. Why I divide into two subtopics? Because in my CV also, my research is almost in fermented food, especially in animal product technology, milk, fermented milk, and fermented meat. And the other one, I would like to share about the non-fermented food. So we move to the subtopic one. It's about the fermented food. What is the fermented food? What is the fermentation? How the beneficial function of the fermentation process to increase the nutritional value of the products. So I would like to talk about this. And this is the right presentation after Professor Azwang, because in our slide also include the application of SDS page to show the beneficial peptides to give our health function. Now, we know that Indonesia is high market value for food industry due to the population of Indonesian people, now still increasing. Compare between our neighbor country, Malaysia, also Singapore, as Indonesia the biggest population. And the second one, if we look to the biodiversity of plant and animals, so Indonesia leads to wide the animal or plants. So the microorganism diversity is also run to this animal and plants. So it's very interesting. So many people, many researchers from another country come to Indonesia, focus in what is the diversity, specific microorganism in Indonesia. For example, the microorganism from Sumatra may be different compared to the Nusa Tenggara and etc. And how about the traditional food? We know that Indonesia has many diversity, food diversity. If we go to the Bandung, so we can find siomay maybe. If we go to the Bali, we find betutu chicken. So also in Malaysia, if we go to the Sabah, this is the special food in Sabah. is different compared to the Kuala Lumpur, also to Penang, also in another country. So thanks to God to give many food diversity. And what is the importance, the food diversity to the microorganism? So it's very important because the I say, I say that the many food in Indonesia is fermented food. For example, tempe, also maybe another food processing. Yeah? It's very contain variety of microorganisms. And we look like to the animal-based traditional fermented food in Indonesia from 3 islands. You can see from Sumatra, we can find dade. Dade is fermented milk, origin from West Sumatra. Also, this is different with fermented food origin from South Sulawesi. In the South Sulawesi, we get danke. Danke is look like cheese, fresh cheese. This is also fermented or not fermented, yeah. It's debatable, but in our research, we could isolate many lactic acid bacteria from the danke. So I can say this is fermented food. In the other island, in Bali, we can find urutan sausage. This is different type of fermented meat, not milk. So Indonesia has many traditional food based on fermentation process. So before we search for the specific fermented food, no. What is the fermentation? The fermentation is an aerobic cellular process in which bacteria can be yeast or other microorganisms convert organic food into the simple compounds from protein to peptide or to amino acid. And chemical energy is produced. But I said that not only an aerobic process, because this is a fermentation with facultative microorganism. In yogurt, for example, this is no strictly anaerobic. We can get oxygen here to produce the yogurt. So this is a facultative fermentation process. In fermentation of food processing, the conversion of food carbohydrates into alcohol and carbon dioxide or organic acid by yeast. Yeast always produce alcohol and carbon dioxide. Bacteria not always produce alcohol and carbon dioxide, but bacteria always produce organic acid. So this is two type of fermentation. Also in the right picture, this is a fermented food that can be used into ketogenic diets. We can see many vegetables here as fermented and then it's very useful for the ketogenic diet. So two-type fermentation. I said before that we have two type fermentation: alcoholic fermentation and lactic acid fermentation. We can see in yeast use glucose and then enter to the glycolysis process and produce carbon dioxide, also ethanol. So if we use yeast for the fermentation process, it is called heterofermentative process. Heterofermentative or heterofermentation because it produces carbon dioxide, also alcohol. The other side is homofermentative process. Homofermentative process mainly used by lactic acid bacteria. So the bacteria use glucose to the glycolysis process and only produce lactic acid, no carbon dioxide and no ethanol. So this is the heterofermentative and this homofermentative. It's very important to we must know because we can select the microorganism, the right microorganism to produce the product that has special flavor, special texture, if we want to produce it. This is the another fermentation food product and their uses, the carbon dioxide in many food, not only animal food, bread, making using bakery yeast. We always use Saccharomyces cerevisiae to make bread, and the alcohol to produce wine. Also, we always use yeast, for example, Saccharomyces cerevisiae also. And the lactic acid is a very beneficial microbes. That is, lactic acid bacteria ferment milk into products as yogurt. Not only to milk, but also in the vegetables, like sauerkraut, fermented cucumbers, and kimchi from Korea are most the studied lactic acid fermented vegetables. Also in Indonesia, tempe. Tempe is also fermented soybean with use bacteria. So the origin of some fermented food comes many years ago, before Christ. For example, mushroom for China, soy 3,000 before Christ from China, Korea, and Japan. Also wine from Europe, fermented milk also from Middle East and China also. Cheese, beer, bread, fermented milk also from Middle East. Fish sauce, pickled vegetables, and also tea. Remember that tea is fermented leaves. So this is the main benefit of fermented foods. Why we eat fermented foods? We can find enzyme. We can get enzymes. To increase enzyme contents helps to absorb nutrients, reducing the need for vitamin and supplements. And why we eat fermented food? Because we can get also probiotics. I will explain more in the probiotics topics because it's the main of our field, of my fields. These good bacteria also can balance in our gut and add the digestion and immune health. So in the pandemic, please consume probiotics, fermented milk, like yogurt, for example, Yakult or another fermented food. How about the safety of the product? The lactic acid during the fermentation process kills E. coli, the pathogenic bacteria. So it is more safe. It is safer to consume than raw vegetables. It's useful also for the preservation products. The lacto fermentation process stores food longer than canning without depleting nutrients. You can see in the can, this is vegetables, also fruits. It's fermented product. we can also pickle products. The other one is nutrition. The fermentation process increases the nutritional value by enriching certain nutrients. In our slide also, we can discuss about what is the health beneficial for our health of the fermented milk. So I will focus in the lactic acid bacteria because lactic acid bacteria can be used to ferment or culture food for at least 4,000 years. For example, yogurt and cheese. This is the homofermentative and heterofermentative. We have been discussed before. This is the shape of the bacteria, cocky, and also the bacil. Okay, what is the probiotics? I said before the fermented foods include probiotics bacteria. So the WHO defines that probiotics as live microorganism. This is very important. Live microorganism, which upon ingesting in certain number, only in certain number, confer a health benefit on the host. On the host, I mean human. This is the bacteria can be included into the probiotic lactobacillus. We can know lactobacillus bulgaricus, lactobacillus acidophilus, and lactococci lactic, and propionibacterium. I haven't used this for the probiotics. And Streptococcus thermophilus, this is the specific bacteria that can be used into the yogurt. Bifidobacterium, if we buy the yogurt, so in the can of the yogurt, this is added by Bifidobacterium for increasing your gut. This is also the probiotics. And I have said that this is the Lactobacillus bulgaricus. So FAO and WHO state the requirement for the probiotics. Most of good bacteria belongs to lactic acid bacteria, yet not all of them are probiotics. So if you find Lactobacillus acidophilus for everywhere, you cannot say this is the probiotics. You must check if the bacteria have for requirement, for characteristics, so it is included into the probiotics. If not, there is no probiotics. So the first one, they should be generally recognized as safe of grass, species, and strain identification. So the molecular technique is very important in the microbiology technique because only for the from the molecular technique we can find strain identification. We can use PCR, also gene sequencing, the specific gene 16SRNA, we can use it to identify the bacteria. And the second one, they should be tolerant to acid and bile. Why? Because remember that good bacteria should pass our stomach. So stomach, very acid properties and also bile characteristic in our JE tribe. So this is the three, they have antagonistic activity against pathogenic bacteria. We will discuss more in another slide. And they could adhere in intestinal cell line. It's very important for the probiotics. Because of this importance of this lactic acid bacteria, so we should explore more lactic acid bacteria based probiotics from indigenous resources, origin from Indonesia, is very important. So this is the effect, the positive effect of probiotic bacteria and yeast. Remember, probiotic is live microorganism that we consume. So this is live microorganism. So they can enter into the gastrointestinal tract. And this is the green one is probiotics, the yellow and commensal. Commensal has comments of probiotics, comments microbes in our gut. And this is the pathogenic bacteria that can cause the diarrhea or etc. So the probiotic bacteria can enter our GI tract, and then they produce some antimicrobial that can attack the pathogenic bacteria. So the pathogenic bacteria can destroy further from probiotic bacteria. And then the other ones, the probiotic bacteria can adhere into the epithelial cell. This is the epithelial cell in our GI tracts. And then they produce immune cells, such as defensins. So the lactic acid bacteria can improve our immune substance, such as defensin. And then this is the defensin here in our mucus. And then the other one, the probiotic bacteria also can interact with the GI tract and then also improve the B cell to produce immunoglobulin A. Immunoglobulin A is very specific, only specific immunoglobulin type in our gut. So it's very important. Next slide is about microbial fermentation as an effect on the host functional properties. This is the picture of how important the probiotic bacteria. The other one, probiotic bacteria also can adhere into the mucus and then improvement the gut system. And then I said before, effect to the B cell to produce immunoglobulin A. And then also probiotic bacteria can produce short chain fatty acid. Short chain fatty acid also can enter to the goblet cell in our mucus, in our intestine, and they produce mucus more here. Also can trigger the T cell. T cell, another type of our immune cell. B cell and T cell. B cell produce immunoglobulin. In special, our GI tract is immunoglobulin A. And T cell, another function of T cell, this is dendritic cell. So it's very important for the immune system. So if we consume probiotics, it is better to consume prebiotics. Prebiotics is carbohydrates that can support the growth of probiotics. We have banana, we have asparagus here, and Indonesia has many sources of prebiotics. This is tempe, burger tempe, miso from Japan, also yogurt and kefir. This is for our health. So, fermented food also can be used biofortification as alternative strategy for improving nutritional status, especially for children. So if we have kids, please um, supply the more of fermented milks like yogurt drink or Greek yogurt is better to be consumed by our kids. So this is the fermentation and the fermentation. What is the fermentation? Definition of fermentation. Also the importance of the fermented product. So in this slide, we can get our specific traditional fermented product from Indonesia. This is dade. Dade, an Indonesian ethnic fermented milk from West Sumatra, is made by pouring fresh raw, unheated buffalo milk or sometimes cocoa milk into the bamboo. This is bamboo into the bamboo. Fresh raw unheated milk into the bamboo with banana and capped with banana leaves and allowed to ferment at room temperature for 2 days. So we can get this product like this. This is dade. And dade has higher nutrient contents compared to the fresh milk because the protein can be added from the bacteria. The other one is danke. Danke is an Indonesian ethnic fresh soft cheese. Fresh soft cheese that is usually made from fresco or buffalo by farmers household in Nrekang Agency, South Sulawesi province. So it's very famous in Nrekang. Danke made by heating with small fire to boil it, and then add sometimes papain enzyme from papaya, resulting in natural clotting, coagulating of the casein, which changes the texture, and then I think the flavor also. The cause of buffalo milk becomes solid due to the separation of protein and water. So this is specific traditional fermented milk also from Sulawesi. And our research has been produced many lactic acid bacteria can be isolated from danke, especially Lactobacillus fermentomycin. So another one, we can get yogurt. Yogurt is very famous fermented milk. Control fermentation by milk by 2 species bacteria. This is very specific bacteria, Lactobacillus bulgaricus and Lactococcus thermophilus. We can say also Streptococcus thermophilus. It's the same type of the bacteria. The lactose is fermented to lactic acid, and it is the one which causes the characteristic card to form. And the Streptococcus thermophilus bring the pH of the milk down to 5.5. And the function of the Lactobacillus bulgaricus convert lactose to lactic acid. Then Lactobacillus bulgaricus also produce proteolytic enzyme that break down milk protein into peptides. So in yogurt, we consume not only protein, but many peptides. So it's very important. Yeah? These peptides also stimulate the growth of the thermophilus. So this is the mutual combination, the bacteria, which produce also lactic acid. So this is the yogurt. Many types of yogurt, set yogurt, stir yogurt, drinking yogurt, and flavor yogurt, Greek yogurt, very solid yogurt, and also frozen yogurt like ice cream. So this is kefir. Kefir is very popular also in Indonesia right now because kefir has many beneficial function of our health. So we discuss what is differences between dade, between compared to the yogurt and kefir. Dade mostly raw fermentation process without addition of specific lactic acid bacteria. So this is uncontrolled fermentation process. That yogurt is controlled fermentation process. We add two bacteria, lactic acid bacteria, Lactobacillus bulgaricus and Streptococcus. And the other one, this is the type, another type of fermented milk is kefir. Kefir produced by grain kefir, not only one bacteria, not only two bacteria. This is the symbiotic bacteria and yeast also. We can find Candida kefir, Streptococcus, Thermophilus also, Acidophilus, also Rhamnosus, another one, Fermentum also, and then Saccharomyces cerevisiae. This many bacteria grow together and form the grain. So the grain is used as starter. The kefir is beverage produced by the action of lactic acid bacteria, yeast, and acetic acid bacteria on milk, which produce distinctive fermented milk with unique properties. So, because the kefir use yeast, so the final product contain alcohol. This is the specific of kefir. The mouthfeel of kefir described to be prickly and sparkly. This is the kefir grain of kefir. It's very easy to produce kefir. We can use the kefir grain and add to the pasteurized milk and then store in the room temperature until 1 day, and then after 1 day we can get the kefir, and we can get also kefir grains to be used as starter to another production. So it's very easy. So this is cheese. I took this picture because it's very interesting time for me. That in Netherlands, I also visited the cheese production house. This is 5 years fermentation. This is the cheese, yeah. For 5 years fermentation process. So this is cheese. We can find at Swalayan many type of cheese, Gouda cheese, also mozzarella, also cheddar cheese. It's different type of cheese. Okay, cheese divided into two technology. It is fermented milk product, and another one not fermentation. So I include into the fermented because many production house using lactic acid bacteria or another bacteria to produce this product. So cheese created by allowing milk, skim milk also, and milk fat solid from the top, and leaving only the liquid protein portion, the whey. So the whey is the byproduct from the cheese production. The types of cheese, unripe and ripe, and also processed. We get the cheddar cheese, that salted cheese. Another one, we get cottage cheese. It's unripe or fresh cheese and ricotta cheese and so on. So in this talks today, I would like to introduce also the beneficial function of the addition of probiotic on fermented food. In our laboratory, we have two or three strain of probiotic, Lactobacillus fermentum and Lactobacillus acidophilus with different strains, and we use it into yogurt and fermented sauces. So in many research, I and Pak Cahyo, also Ibu Dr. Zakia also doing this one, also Pak Resta, also Pak Happy. We combined the probiotic bacteria to improve the health beneficial function of the product. Altogether, we use, like I said, bacteria Lactobacillus plantarum and Lactobacillus fermentum to the yogurt, and then we get some function properties. The first one, anti-hypertension. The second one is anti-diabetic, and the third one, and hypocholesterol. It's the publication of our research. The first one, antimicrobial activity, also the anti-hypertension. This is anti-diabetic. So this is a part of the research. Activities of ACE inhibitor enzyme on probiotic yogurt. ACE inhibitor is enzyme that contribute to hypertension. So we can see that, okay, this is the mechanism in our body. So the angiotensinogen is produced by the liver and then convert to the angiotensin 1 by renin that can be produced by the kidney, and then the angiotensin 1 convert to the angiotensin 2 with angiotensin converting enzyme that proposed by the lungs and kidney. So if we have the product that can inhibit this one, the ACE and the angiotensin converting enzyme, so the angiotensin 1 cannot be converted to angiotensin 2. So the hypertension is reduced because the angiotensin 2 increase tubular natrium chloride reabsorption, and also the vasoconstriction in arteriolar, increase in blood pressure. If we can find the substance, this is the peptide, the specific peptide that can inhibit this enzyme, so the angiotensin 1 cannot be converted to angiotensin 2. This is the mechanism. I back to this one. We use goat milk yogurt, yogurt probiotic, and yogurt probiotic roselle. We use Lactobacillus plantarum for the probiotic. So the research, we found that all the yogurt added with probiotic can inhibit the enzyme. This is the value. So IC50 value was defined the concentration of inhibitor required to inhibit 50% of the enzyme activity. So this is the yogurt probiotic. We add roselle flour extract, and we find that the yogurt probiotic roselle is better than yogurt probiotic to reduce hypertension. Another function, why it can inhibit the enzyme, because the fermentation, this is SDH profile. Remember the slide or the lecture from Professor Arts one before, the SDH page electrophoresis to detect the molecular weight of the protein. So this is yogurt probiotic, and then yogurt probiotic with roselle. We detect the molecular weight of the peptide of 3 products. We can find this one, yogurt. There is alpha casein here, and then yogurt probiotic. This is the yogurt probiotics with roselle. And this is the peptide below than 10 kilodalton. So we can see in this profile that yogurt with addition of probiotic that produce low peptide more than only yogurt. But if we add by roselle, it is better than yogurt probiotic, also yogurt. Low molecular weight of peptides were agent for ACE inhibitor. So many research can be found the sequence of peptides that can inhibit the ACE. Okay. The other positive function of probiotics that we use to be a starcher culture in yogurt, also for cholesterol assimilations. We use, this is yogurt with roselle, this is yogurt roselle with probiotic Lactobacillus plantarum, and the last one, yogurt roselle probiotic Lactobacillus fermentum. This is our researcher, our students, Burhan 2017, found that cholesterol assimilation of 2 strains of probiotic Lactobacillus fermentum can assimilate cholesterol 17% and fermentum 33%. So the probiotic depend on the strain. I mean that Lactobacillus fermentum maybe can use better for the anti-diabetic, but fermentum is better for the cholesterol assimilation. So it's very interesting to study about the probiotics. Yeah. So we can see here that the yogurt probiotic with Lactobacillus plantarum has highest cholesterol assimilation compared to the yogurt roselle, also this one, fermentum. But the stability of fermentum is better than plantarum. It's very interesting research also, but we don't continue this research because there is no student interest this. So this is another mechanism. The first mechanism is assimilation. The first mechanism is cholesterol assimilation. If we consume the food with high cholesterol content, so the probiotic in our gut can absorb the cholesterol from the food and then excrete to the excreta. So the cholesterol cannot be absorbed to our body. So the level absorption cholesterol in our body is lower. So this is the first mechanism. The another mechanism that is the probiotic has bile-salt hydrolysis activity enzyme. Probiotic produce enzyme, bile-salt activity enzyme, and then contribute to cholesterol metabolic pathway. Bile-salt hydrolysis enzymatic activity hydrolyze conjugated bile acid to deconjugated bile acid. This is it. Conjugated bile acid can convert to deconjugated bile acid, then deconjugated bile acid and cholesterol will excrete to feces. So this is we use lactobacillus. So lactobacillus can maintains the cholesterol absorption in our body. The last one is the function probiotic as anti-diabetic. This is many mechanism and very specific mechanism. So we still continue this research, maybe by support from the students. The activity of enzyme alpha glycosidase inhibition of yogurt. Alpha glycosidase is an enzyme that plays an important role in the absorption of glucose in our gut. So if we consume food that have high glucose level, so the alpha glucosidase enzyme in our gut can support the absorption of the glucose. If the enzyme can be inhibited by probiotics, so the glucose cannot be absorbed in our body. This is the result. We use yogurt as a control and yogurt probiotic with Rosel. So the result showed that the probiotic yogurt supplement with Rosel extract has the highest alpha glucosidase inhibitory activity among all treatments with 37% inhibition, but it decreased during the storage. So this is a very important thing that we can use probiotic as main probiotics products. We consume probiotics with natural probiotics that no addition of sugar, yeah, but it's very acid, yeah. This is another mechanism of probiotic function in glucose metabolism. The probiotic produce short chain fatty acid. It effect into our intestine, and then SGFA can absorb into the systemic circulation that can be a positive impact to the increasing insulin sensitivity. The other one also in the muscle also increase insulin sensitivity. So this is another mechanism. Okay. This one for the fermented foods. This is based on our publication with Pak Cahyo and Ibu Zakia. we why we are interested in fermented sausage, because in Indonesia has fermented sausage, traditional fermented sausage from Bali, urutan. Urutan is a Balinese traditional fermented sausage made from lean pork and fat mixed with spices, sugar, and salt. So this is uncontrolled fermentation, spontaneous fermentation. We have no addition of the Lactaceae bacteria. Lactaceae bacteria can be applied to control the quality of the product, and we can change the source of the meat from pork to the cow, from to the cattle, or maybe red meat or the poultry, from poultry. So Lactaceae bacteria are responsible for lactase production, for the flavoring of sausage, also contribute in nutritional value that are produced during the fermentation. What is the importance of fermented sausage as functional food? The first one, inhibition of intestinal pathogenic bacteria by production of the organic acid and by low pH value, and to prevent of pathogenic adherence to the intestinal mucosa. As I mentioned before, the production of bacteriocin. Today I am not explaining about the bacteriocins, and the fourth, increase of food assimilation, and the fifth, immune stimulation. And how about the products? Advantage of probiotic products: quantity and quality of amino acid content of the product are increased. Better profile MUFA and PUFA, polyunsaturated fatty acid, and save life as longer at room temperature. So this is the picture of fermented sausage. I took this one in Netherlands. This is fermented sausage. This is yeast, It's very interesting, like tempe in Indonesia, I took this one in Australia. This is in Australia. This is the meat store in Australia. So you can see, free range eggs. This is natural eggs. It's very expensive eggs because of animal welfare, So this is in traditional market in Adelaide. It's our dream, to produce urutan more international product. So before to the next subtopic about the food, fermented, non-fermented food. So I would like to stop now, and I invite the students who would like to give question. Pak Cahyo.
Okay. Thank you, Prof. Irma, for the presentations. And I think the students are impatient to ask the questions. I received already several questions that text to me privately and also from the chat room. I will go first from the chat room.
If you can answer, please answer. So I can drink now.
I will play my role as moderator, Bu.
No.
Okay, I will go first to Gilang. Gilang, are you there?
Yes, sir.
Okay, Gilang, please convey your questions directly.
Okay. Right. Good evening, Prof. Irma. My name is Gilang from IBB University. I have a question about Indonesia fermented food. Are the bacteria for fermented food is origin from Indonesia, or there is introduction bacteria from other countries such like China or Dutch? Thank you.
Okay, Bu.
Okay, Okay, the comparison between the bacteria isolated from fermented food from Indonesia compared to the other country. That is right, Pak Cahyo?
So the bacteria that you use, they are solely originated from the Indonesia, or you also use the bacteria from the other countries like China or Netherlands and so on?
Okay. Sorry, yeah, I cannot reach your question. So we use local lactic acid bacteria that has been isolated from fresh meat. From fresh meat, also from dangkel. Dangkel is fermented cheese from South Sulawesi. We can get Lactobacillus fermentum, and from fresh meat, we can get Lactobacillus plantarum and Lactobacillus acidophilus. So it's very different compared than lactic acid bacteria isolated from another country.
Maybe Bu Irma can also elaborate the Lactobacillus plantarum that you have. Are they different with the Lactobacillus plantarum from other countries? Or
please answer, Pak Cahyo. Very biotechnology technique that we use. So I and Pak Cahyo is a very strong partner to be a researcher here. Yeah. So Pak Cahyo, please answer.
Okay, I think it's answered already. All right, okay. We go to the next students. Maulita Putri, are you there?
Sure.
Okay, Maulita. Is your camera has a problem?
Yes, I have some trouble with my camera.
All right, okay. So then proceed. We can get your voice clearly.
Okay. Good afternoon, Prof. I'm Maulita Putri from IPB University. And well, I want to ask about how is the process of drying fermented products such as yogurt, whether during the process the bacteria contained will die or not. Because I have a yogurt product and I want to try drying it, but I'm still confused. Thank you, Prof.
Okay, that's good question, Smolia. Thank you. Prof. Irma.
Okay, there is further technology to produce freeze dried, produce yogurt, fermented milk, freeze dried yogurt, yeah, because the process is include the encapsulation process. So the bacteria is encapsulated with encapsulation agents, and then the bacteria enter to the spray drying or freeze drying, but the bacteria can be saved with the encapsulate with specific agent, I think. So there is the live microorganism.
Okay, thank you, Prof.
It's a specific technique, yeah. But if you would like to do a research in this encapsulation of lactic acid bacteria, it's open to you, yeah.
Okay, thank you, Prof.
Yeah.
Maulita, just curious, are you now doing your final year project, your scriptio or your thesis?
I'm sorry, I'm in the third in this semester, but I have a product. There's yogurt. There's some production of yogurt.
All right. You have your own product.
Yes, sure.
So you are the entrepreneur.
Maybe soon.
Okay. So what Prof. Irma suggests is you cannot dry directly. You cannot drying the product directly, but you have to firstly encapsulate the starter and so on.
Yes, there's
Please
higher
consult
technology.
with Prof. for the technical advice on your product.
Sure.
Right, we go to the next students. Yeah. We go to University Muhammadiyah Malang. We have Carlina. Hello, Carlina.
Yes, sir.
Okay. Could you please convey your questions directly?
Good afternoon, ma'am. My name is Carlina. I'm from University of Muhammadiyah Malang. My question is there is any possibility while the food on a fermentation process but contaminated with other bacteria?
Okay.
Will there be any physical changes?
All right. Okay.
Yeah, thank you.
Okay. Thank you, Kalina.
Yes, we can use 2 or 3 lactic acid bacteria together. This is a symbiotic bacteria that can be used, but with the specific function. For example, we can add the Lactobacillus bulgaricus, Streptococcus thermophilus with bifidobacteria or Lactobacillus acidophilus all together. Lactobacillus acidophilus, and then we can also use Saccharomyces cerevisiae, but Saccharomyces cerevisiae is yeast that produce alcohol. Yeah, this is main constraint in our food production for halal issue.
I mean, like a pathogen bacteria.
Yes. So maybe if we make a fermented food, Burma, using probiotic, does it mean that we won't have any contamination from other pathogenic bacteria?
If we use the right lactic acid bacteria, so that can produce antimicrobial substances that can inhibit the pathogenic bacteria. So if we get our product contaminated with pathogenic bacteria, so we can conclude this fermentation is not success.
And if it's contaminated, the characteristic of product also will be affected, right?
Yeah.
Okay.
The
Kalina, is it clear?
Order.
Yes, is it clear? Thank you, ma'am.
Okay, good. Then I think we move to Australia. We have Sam. Hello, Sam, from Western Sydney University. You there?
Yes, good evening. I'm here.
All right. Please, Sam, I think we are glad to have your questions.
Thank you. Good evening, Professor Emma. Now, my question is regarding the percentage of the ACE inhibitors. Now, what's this percentage related to? And I understand I'm probably not very familiar with the ACE inhibitors, but yeah, what's the percentage related to? What's the percentage of?
Where is it clear?
Okay. Thank you, Sam. It's very good question. The percentage is describe the activity. So if the 100%, so this is the maximal activity to inhibit 100% can inhibit ACI. This is the angiotensin 1 cannot convert to the angiotensin 2. But in our result, only 20%. So the probiotic bacteria that we have only reduce 20% of the this enzyme.
So okay, well, I understand the higher the percentage, the better it would be, right?
Yeah.
Okay, so okay, thanks very much for that.
Okay, thank you, Sam. All right, okay. We move to Masita. Masita, are you there?
Yes.
All right, Masita.
Hi, Prof. Irma.
Introduce yourself.
Hello, Masita from Malaysia, right?
Yeah, from Malaysia.
Okay.
University Putra Malaysia.
Okay.
So I have a question regarding the probiotic and also as animal feed. So what type of probiotic that we can use? I mean, in like our climate, Indonesia, Malaysia, that are very useful as a feed sources for small ruminants during drought.
Okay.
During dry season. Thank you.
All right. Any specific probiotic that we can suggest for the small ruminant, particularly for during dry seasons?
I cannot answer well because I'm not a nutritionist. Yeah. So I'm not I am sorry about this. Maybe if I know that the probiotic can be used for the feed, should digest polysaccharide, yeah, cellulose maybe, cellulose, yeah. But I cannot answer well because I am not a nutritionist. Maybe Pak Cahyo can you help me?
I have no idea also. I have no idea. But yes, probiotic can be used, but which type is the best one? I have no idea. We have to consult to Prof. Komang.
Yeah. I'm sorry, Bu Masita.
All right. Okay. Any other questions from the students? I have the written one. Burma related to the cost when we apply the probiotics in the, let's say, milk fermentations. Is that will it affect the cost of productions and price of the product?
In where side? I mean that if we are producer, so it will improve our price of our product. If we are consumer, so this is the high price, but we can get more nutritional value and beneficial health of the product. So it's a choice, I mean, yeah.
Okay. Okay. All right. We have more questions coming. We go.
But I have more slides, but
okay.
maybe this is the 5 p.m.
So how many slides still you have?
Don't know. This is no non-fermented product, yeah.
Okay, because there are several questions remains. If I think it's good to answer that questions because the students are very interested in your topic. And then after that, let's see if we agree to continue the presentations and so on. All right. From what I cannot see the name. Pram, are you there? Hello, Pram. All right, I will read the questions. Pram, do you prefer to ask directly?
Hello, good afternoon.
Okay.
I'm sorry, I couldn't turn on my camera.
All right, okay, proceed.
My name is Pram from IPB University. I want to ask about dadi. As far as I know, to make a dadi, we use local bacteria from the bamboo to do the fermentation. Please correct me if I'm wrong. My question is, could we make dadi, but without using the bamboo? We just take the bacteria from the bamboo and put it directly into the buffalo milk.
Yeah.
Thank you.
All right. Okay.
This is a specific name of the dade as produced by bamboo, yeah. So we can add the lactic bacteria to control the fermentation, but many research results.
Hello. Bu Irma is freeze. All right. Okay. Okay, Pram, I think while waiting Bu Irma to back to the Zoom, I maybe can help to answer. I did this research during my for my undergrad thesis. We make a dadi like milk fermentations using without using bamboo, but we add just probiotic that previously been isolated from the original dadi. And yes, it can the result. The result of the trial produced a similar product like dadi, but I don't think if you sell to the market, what people understand about dadi is the one that package it in bamboo, not the one that package it in the plastic what is that, can or something. So yes, basically we can make it without using bamboo, and in fact, by using bamboo, there are some disadvantages on this because of some other contaminant pathogenic bacteria coming from bamboo and affecting the quality of the products. Well, many research on that one actually, but I'm talking from my own experience when I think on 2004 when I did this work. All right, Bu Irma is back. All right. Okay, Bu.
No, maybe my signal.
All right, okay.
I have one question from Yolandani. Yeah. Can I answer this? Thank you, Yolandani. I have a question about how to minimize the contamination. Of course, we can use specific lactobacteria that produce bacteriocins. That bacteriocins is only the perfect, the best antimicrobial substances that can inhibit pathogenic bacteria.
Okay.
Yeah. So you can select lactobacteria that produce bacteriocin.
Okay.
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 Can I?
I have two more, Bu. But before that, I would like to call Rafli Lukman. Rafli Lukman, are you there? Rafli? Hello, Rafli. Is not here? Sleeping?
No. In the dark room. Rafli.
Hello, Rafli. Okay. Anyway.
Rafli.
Is he here? No. All right. Okay.
No. He is here.
He's here. Okay. Rafli? No, he's not answering. Okay. I will call back Rafli later. I go first to who is he? I don't know the name. It's but he said that she cannot ask directly or Asifa from IPB University. She cannot ask directly because the microphone has some problem. The question is, I used to try to make homemade yogurt using fresh milk and leftover Greek yogurt. However, sometimes I'm not successful in making it, although I have made sure the container is clean and tightly closed. Do you have any suggestion so that the yogurt can be successfully made? Or should I use yogurt starter in order to minimize production failure? So it seems like the question is actually, if we make yogurt, do we really need to put the starter, or we can just put the milk on the can and leave it overnight and become yogurt?
No.
What about that, Prof?
No, there is no yogurt. Yeah, yogurt is produced by two bacteria, Lactobacillus bulgaricus and Streptococcus thermophilus. But if you would like to produce successfully, please train and come to our laboratory. Because many specific technique, to produce this.
Okay.
Okay.
All right. You can just leave your milk, on the table and so on, and then you expect to have yogurt, Instead, you will have your milk become deteriorated because of contamination. All right. Okay. Bu Ir, you still have some more slides, right?
Yeah, maybe 10 minutes,
All right. Okay. The student, is it okay we continue for the presentation, More 10 minutes, about 10 minutes more. While waiting, Rafli, you there already?
Can you see our slide screen?
All right, yes, Bu. Please proceed.
Okay. Everybody, thank you for your interesting question. And this is 5 p.m. My time is out actually, but 5 minutes we will discuss about the non-fermented food, and we will discuss more if you want interested in. So we can discuss by email, non-fermented food. I explain quickly about the milk. This is the milk, We drink milk with high protein compounds, high protein contents, and vitamin D. We drink pasteurized and homogenized milk. This is the nutritional content of milk: vitamin D, vitamin A, minerals. This is the milk products. So many milk products can be produced from the raw milk. Types of dairy products. Thank you for Dr. Arah. This is the presentation from the Dr. Arah. Milk, cream, and then butter, cheese. This is the cream. cream is a more concentrated form of milk, whipping cream. Yeah, we can use it to the maybe to the another food. Yeah, this is frozen ice cream, also the frozen yogurt. This is the meat product. It will plate. Yeah, so the high biological value of protein. This is meat, important micronutrients. Most healthy, balanced diet will include lean meat. Our colleague, Pak Heri, has been explained about the dendeng. Dendeng, yeah, many meat products from Sumatra. This is also the dendeng batoko is typical West Sumatra meat products because in the processing technique we use mortar, so the meat becomes soft. Yeah. The another products from East Nusa Tenggara province, we call it sei meat. This is cooked by firewood with the hot smoke. This is the sei meat. Another one is shredded meat. This is the dried meat. It look like the cotton fibers because it is dominated by dried muscle fibers, and because it is dry and has almost no remaining moisture content, so it can be stored for weeks to months. Another traditional products is rendang. I won't explain more because this is the Pak Heri can explain well. Okay, to the another product is salted eggs. It's come from China, I think, yeah, but adapted to Indonesia. The another one is also pindang eggs. Pindang eggs sometimes are sold as snack, yeah. This is a processed milk that avoid using certain herbs that they have a distinctive taste, aroma, yeah, and appearance. Also, the last one is honey, yeah, because the consumption of honey is a very long history. This is the honey. Honey is naturally liquid, generally has sweet taste. It's better than sugar, of course. In Indonesia, the average production estimated to reach about 4,000 tons, yeah, per year, maybe more now, yeah. And every area has specific honey, but I am not an expert in this field. Maybe another expert, another lecturer can explain more detail about this, Ibu Dr. Yuni. I will more detail about what is the function of honey. Yeah, honey contains a lot of nutrients, including protein, vitamin, mineral, and in the pandemic COVID-19 now, so it is better to consume honey compared than sugar. Yeah, this is the nutritional value of honey compared to sugar. Yeah. We can get more vitamin C, riboflavin. Yeah, there is very useful for our health. So it's my end presentation. Thank you very much. You can contact me by Facebook, by Instagram, also by email. Thank you very much.
Okay, thank you.
Thank you very much.
I think I will continue with the questions from students, maybe 2 or 3 more questions, and then before we end up the sessions because we are behind schedule already. Anyone who has the questions, just chat me privately or put it in the public chat room. I have some questions for that left. I haven't conveyed yet. Riza, are you there? Okay, Riza Maharani. Okay, Riza, would you like to proceed with your questions, please?
Yes, sir. Good evening. Is that clear?
Yeah, it's clear. Yes.
So I want to ask some questions, Professor Irma. The first question is, what makes yogurt have so many variation product? I mean, like you explained in your presentation before that yogurt can be stirred, drink, and etc. So is it because of the procedure or number of bacteria or even the temperature? And then the second one is it possible to change yogurt to a different one? For example, I've got a drink yogurt, then I want to change it into stirred yogurt. Is it possible to do that? And then the third one is there any differences in processing fermented sausage between pork and cattle? Thank you, Professor.
Okay, so there are three. Wow.
Many questions, yeah.
Okay,
Sure, this.
You want me to repeat the questions?
Okay, the first question
Right.
is about the type of yogurt, yeah. So
Why there are so many type of products? Why there are so many type of yogurts? Is it because of the type of bacteria that we add are different, or because of the temperature, or so?
This is the technology process is different. Set yogurt is different with stir yogurt, also Greek yogurt, but also the components, the materials we use. The Greek yogurt, we can add the sometimes polysaccharide, yeah, to more the texture more very firm.
Okay. All right. The second one is it possible to change the yogurt from one form to another form? Let's say from drink yogurt to stirred yogurt.
No, this is the final product.
This is final product, so you cannot change anymore.
Yeah.
Okay. Last one is if you make fermented sausage. I think this is your field.
Yeah.
Pork sausage and beef sausage will there any difference on the products?
No difference, yeah. But urutan, the traditional fermented sausage from Bali use fresh intestine as a casing, but we can change it with the artificial casing. I think it's enough, Pak Daya.
Right. Okay. Thank you, Prof. Last questions, Bu. Sorry. There are many admire. Okay. Mikhail, are you ready? All right. Please proceed.
Can you hear?
Yeah.
Thank you for the lovely presentation, Professor Irma. I want to ask several questions. So a friend of mine is not comfortable drinking milk because he felt like vomiting after drinking milk, and for me too, if I drink milk, I got sorry to say, like menstruate. And is there any way for us people who aren't used to drink milk so that we can consume milk? Yeah, and the second question is adding baking soda affect the microbial activity in milk? Thank you.
First question, yeah. The first question, please add flavor in your milk, yeah. Sometimes you can add fruits, many fruits. Also, you can add maybe also vegetables, yeah. Also flavor, if you like flavor banana, so you can add a banana. If you like orange, so add orange. It's depend on you, yeah. And also sugar, but the percentage of sugar is I think don't more than 10%. It's the question one, and the question two I cannot get Pak Cahyo.
Mikhail, can you repeat your second question?
Yeah, so my second question is about adding baking soda in milk. Will it affect the microbial activity in milk?
Okay. If we add baking soda, will it affect the microbial populations or characteristic of the milk itself?
It will reduce the microbe population.
But I think baking soda has safety issue, is it?
Yeah, yeah, safety issue, but maybe can inhibit the growth of microbes.
Okay. All right, Mikhail.
Thank you.
All right. Okay. Thank you, everyone, for being active during the discussions and also uh, being awake during the lecture. Unfortunately, I have to be clear enough that because many of you will convert the attendance of this lecture for your credit, so we treat you like attending the normal lecture. Yeah? So all right, okay. Anyway, before we end up completely the lecture, I would like to present the certificate appreciations from the committee for our speaker, Professor Irma. Let's see. Okay, so everyone can see my certificate. All right. Thank you, Prof. Irma, for the very interesting talk and lecture on the traditional animal product and processing in the summer course program. And the certification is on behalf of Department of Animal Product and Technology and also Faculty of Animal Science. The talk is definitely fruitful for the students and also for the for all the participants today. Thank you very much.
Thank you. Thank you.
All right. Okay. Before I completely end up the program today, I would like to share you for the students assignments that maybe you can start to think or start to work on it during this weekend. I'm very sorry for destroying your peaceful weekend. All right. For there are several issues here. There are several points here. For the credit earning. The course content comprise approximately 60 hours, which is equal to 2 credit points with detailed descriptions of 20 hours lecture, 20 hours self-study, and 4 hours allocated for final assessment. And then students will gain 2 credit points with the subject name of summer course on animal productions after completing the final assessment. So if you don't do the final assessment, you won't get any credit. Yeah. For the other university, I think there is a procedure to transfer the credit. I think you can contact the committee about the procedure for the transfer of this credit to your courses. Yeah. And then for IPB students, I think the courses you can register directly from IPB mobile, if I'm not mistaken. All right, but so to note that assessment is very important for completing your credit points. All right, the assignment one. For individual, you will have review assignment as been described by Prof. Asep yesterday, following the format that being also described yesterday, and you will have a deadline until 2nd September. 2nd September is on Wednesday, at 12 p.m. So at noon Indonesian time. And then the second assignment is group assignments, whereby the committee has already grouped you based on the university and country basis. And then what the group assignment will be is you discuss about current situations in your district or your country that related to the animal productions, animal product processing technology, animal biotechnology, food science, etc. And then at the end, the group will be requested to present your discussions on 2nd September or 2020. What is this? 2020. Okay, so so on 2nd September, there will be for both individual and group assignments. there will be presentations for group and also the submissions deadline that for the individual assignment. I think that's all. Okay, this is the group grouping made by committee. If you have any issue, let us know. Pak Asep, are you there?
Yes.
Okay, correct me if I'm wrong, Pak. D1 means group 1, is it?
D1 is animal production program.
Okay. I think better you explain directly.
Okay. So I try to continue Pak Cahyo talk.
Yeah,
so this is related with group presentation. D1 is animal production program, and then animal product processing. And after that, we have a food science technology student also, and then we have a from University of Samratulangi group, and then University Diponegoro, University Brawijaya, and UMM. Maybe we can merge together from East Java, and then Jiangsu. Yeah, you have a